But ma'am, it doesn't proceed actually in the next slide.
Okay.
Page down.
Okay, okay, sir.
Yeah, okay. It's working. It's okay.
Is it clearly visible, no, ma'am?
Yeah, it's
It's
visible.
perfect visible.
Yeah.
Okay, okay, sir.
Prosím tě, Agáto, podívej se na to YouTube, ono to tady asi jak běží, ale to je nějaká, teď došlo k nějakému úplně magii, který nerozumím teda.
My teďko slyšíme asi ten záznam, Jirko, od tebe, nebo nejsem si úplně jistá.
No, částečně jo, počkej, já to tady stlumím někde. Jo, tadyhle vypnu mikrofon, dobrý. Vidíš to na tom správném odkazu, že tam běží to na tom YouTube, to teďka se tam vidíme.
Jirko, promiň, ono do toho zase mluví ten záznam a já tě vůbec neslyším. Já teďko nevím, jestli je to moje chyba.
Vidíš to na tom YouTube normálně na té správné adrese?
Ano.
No, tak to běží nějak. Vůbec nerozumím tomu, jak to, že to tam je, ale to je jedno.
Ale je to tam, takže je to v pořádku.
Tak to tam je prostě.
Dobrý, super. Tak jo, já myslím, že snad všechno máme teda doufám v pořádku. Já teda už nazdílím tu moji prezentaci zatím.
No a já pak pustím ještě to nahrávání, až to bude jako to dobrý.
Tak. A teď jsem se ztratila. Tak. Vidíme to dobře takhle, mám to dobře?
Jo.
Super. Ale je to teda nepříjemné, že já vůbec nic nevidím teda. No, tak.
No ono, když si dáš ty dvě obrazovky, tak si to ještě zkus, dej si dvě obrazovky.
Já si to ještě zkusím, ano. Ano.
No, a teďka tuhle ukončí tu prezentaci.
Ano.
A teďka si jako v tom nastavení té prezentace, dej si třeba pustu, jako by to sdílej tu obrazovku.
Ježiš, teď jsem si to zavřela, zase sakra.
Nevadí.
A mám si ji teda otevřít, ale na té druhé obrazovce.
No počkej, to je jedno. Někde si teď tu prezentaci si dej a dej si tam v tom prezentace.
Ano.
Počkej, já se podívám, jak to tam je. Tam je zobrazení, ne, prezentace.
Ano.
A tam si můžeš nastavit, na kterém monitoru se ti to zobrazí. Tam máš monitor.
Mhm.
A tam si dáš, jako, že je to buď to prostě ten druhý, než na kterým běží to, a potom si to nazdílíš, jo?
OK. Já to zkusím teďko.
No.
Jo, ano,
A
skvělý.
teďka si tam dej prezentace, jo, a teďka si dáš no a teď si to zkusí dát full screen teďka, tak? Jo?
Super.
Bingo. A vidíš přitom ten zoom, jo? Trošku.
Ano.
No.
Teď to vidím dobře.
Tak jo.
Mně jde o to, abych zvládla sledovat i ten YouTube, no. K tomu, víš?
No.
Viděla obojí, prostě.
No ale když budeš sledovat YouTube, tak bude někdo přednášet, takže nemusíš si...
Jo, to je pravda. To je pravda.
Jo?
Super.
No,
tak jo,
no.
a já to teda jenom takhle občas teda to postrčím vždycky na... Jo, ale je to vidět takhle celý? Já to totiž teďko na té obrazovce vidím ořízlý.
Ne, já to mám celý.
Jo, dobrý. Dobrý. Super. tak asi jsme
Ale
ready.
proč to běží na tom YouTube, to je mě teda záhada, jako, ale
Ale je dobře, že to běží. Ono je to asi je to prostě nějak skřížený, ne? Teda ten odkaz, nebo...
úplně mě to není jasný, kde jsem... Dobrý. Hlavně, že to běží. Tak jo, já tady budu před 9, tady budu zpátky.
OK, děkuju.
Prosím tě, ještě jednu věc. Zkus tý ženský napsat, ať si ze zoom.us/download nebo downloads, já teď nevím, stáhne tu aplikaci.
Jo, ale ona už je připojená. Už mi psala, že je to v pořádku, takže já doufám, že...
Tak jo. Kdyby někdo měl problém, tak prostě je Zoom a zoom.us.download a tam z toho, ať ti to dám ještě tady do chatu. Jo, mám někde chat. Ale tam, ať si když tak z toho stáhne tu aplikaci a pustí tu aplikaci. Ne přes browser. Jako, jo, přes ten browser s tím jsou občas problémy.
Jo, dobře. Tak jo, já se teďko taky na chvilku teda odpojím ještě, abych si dostahovala ty prezentace a jsme připraveni snad.
Tak Bartuška není, ale bude, jo. Teď jsem mu volal.
Honzo, všecko OK, jo?
Není tady ještě Bartuška, ale volal jsem mu a počítá s tím, má vést někam dceru nebo co.
Jo, ale ten Petr je tady už, já už ho vidím.
Petr Heděnec tady je, ten Martin tu ještě
I'm
není,
here.
ale bude. A nazdar Petře, vypadáš čím dál tím líp, člověče.
Děkuju.
A tadyhle...
Nevidím pan nebo paní Xia Kang.
To jsem viděl včera.
Kang.
Pardon.
Jo, ta je důležitá, protože ta je hned první.
Mhm.
Kolega Ječka je tady, taky ne.
On se přihlašoval na chvíli před chvílí a možná si jenom odskočil zase, nebo taky ho teď nevidím, ale už tady jako byl ráno.
Byl tady, jo. Bartuška teda by měl být.
1, 2, 3.
Tak ono ještě 10 minut, ne?
8 minut.
8 minut, no. Je fakt, že z té první sekce tady moc lidí není, no. Ten Sangeta, Kopagy taky hodně vidím. A Šneha taky nevidím.
Jo, ta si to už vyzkoušela.
Jo. Tu už vidím. A tohle před ní nevidím teda.
Sanjita Mukhopadhyay, paní, to je taky paní teda. A nevím, taky nevidím.
Je to Sneha nebo Šneha?
To nevím, bohužel.
To je, neudal nějaký folva.
Jo.
Hlavně, že tady chybí ta první, to je trošku problém.
Mhm.
Tak když tak přehoďte pořadí, jako jestli tady.
To přehodně pořadí, no. On ten Bartuška, který je třetí, tak taky jako počítá s tím, že přijde na čas.
Hmm.
Já můžu říct, Sangita může jít místo Bartušky. Jestli.
Jo. No, nebo ta Šneha, ta Sangita ještě nevidím jí. No, ještě 6 minut, tak uvidíme.
A včera to Honzo skončilo nějak v pořádku, jo? Já jsem
Prosím tě, to jako uvedl, pak jsem byl v nějakých těch rumech a pak jsem taky utíkal na vlak, jo.
Jo.
Ale pozval jsem je předtím na tu další sekci a to jsem prostě ještě něco jsem řekl předtím, než jsem šel do těch rumů. Tak snad to jako se ukončilo důstojně.
No, se to možná ukončilo tak, že to prostě skončilo jako vypnutím, no.
No,
Já
tak,
jsem Janě řekl, ať to vypne, prostě, no, tak.
Jo. Tak to asi tak dopadlo.
No, a teď už budou ty roomy pojmenovaný podle těch lidí, takže a budou tam ty lidi přiřazený, takže ty řečníci, takže v tom poslední, já myslím, že to fungovalo líp, jako.
Jo, to už si myslím, že jo. Teď mi přijel
No, tak já to udělám přesně tak, akorát já mám...
jako zase přijde, že je tady méně lidí než před chvílí.
Honzo, je to teda 15 minut, 10 minut přednáška a 5 minut diskuze, jo? Nebo?
Zhruba, no. Oni samozřejmě některý se jako...
Přetahují. Já jsem se viděl včera začátek, trošku přetáhli, no.
Ale tak přiměřeně, no.
No, jako... Jo.
Nemáme žádný paralelní sekce, je dokonce možný, že prostě tady jeden ten řečník jako vypadne.
Aha.
Takže bych se jako netrhal bych si játra, jo, ale zase, aby prostě někdo mluvil jako hodinu.
Jo, a Petře, prosím tě, dotazy teda z YouTube mám dávat jenom do chatu, nebo...
Jo, můžeš mi to když tak do chatu.
Jo.
Když tak, já to nějak uvedu. a jako uvedu tě a pak by se z toho chopil. Jo. Akorát, že teda ta
Šia Kang tu není.
Kang tu není. No. Já si myslím, že jsem ji viděl včera. A ona s tebou komunikovala, ne? Ta
Se mnou ne?
Agátou.
Se mnou? No, nějak zvlášť teda.
Jak zvláště.
Mhm.
Ale jako nějakou respons, jako že je, tak byla, jo.
Jako má přihlášený ten poster a vlastně já to hledám teďko v mailu, ale jakoby nemám nějakou reakci.
Jo, no tak dobře, tak když taky přeskočíme a začneme Jačkou rovnou, jestli tu Jačka je teda. Já mu když tak zavolám.
Hele, nemám teda žádnou reakci, ale nemám ani zprávu, že by to nepřišlo.
Jo, ale já jdu volat. Jačka se jde připojit, počítá s tím, že by když tak začal dřív. A tím pádem, hele, jako nemusíš to nějak hrát.
Jačka už je teda.
Kdo?
Jačka už vidím připojený.
Jo. Ale Lukáš,
Dobrý den, slyšíme se?
Haló, slyšíme.
Slyšíme se, rádi tě vidíme.
Tak já zkusím nasdílet teda obrazovku.
Ještě vydrž chvilku, jo, ale to jako jak uvedeme.
Jo, asi, že si to nechce vyzkoušet, pane Jačka.
Jo, tak si to vyzkoušej. Vyzkoušej si to, prosím tě. To je dobrý nápad.
Jo,
Okay.
já teda vypínám to svoje a vypl jsem to, doufám.
Jo. Perfekt. Jo, vidíme to.
Ctrl L by měl jít full screen.
Je to OK takhle?
Perfekt. Teď je to perfektní.
Máš ještě nějaký další obrázky? Co si zkusím posunout? Jo, výborně, posouvá se to, vynikající.
Perfekt.
Xia Kang is here.
Jo, hele, tak nezlob se. Budeš po ní. Už jsem myslel, že nepřijde, tak jsme tě tady takhle nabuzerovali.
A kde já si to teďka, tak já dám...
Jdeš na to share screen a tam bys měl zase skončit. Jako.
Jo, já ho schodím, víš tak.
Okay.
Jak je ten zelený vprostřed, tam, no dobrý.
Super.
Vidíš ji, tu Xia Kang?
Vidím.
Je tam.
Je tam. Tak jo, hele, tak já něco řeknu. Já začnu. OK. Ladies and gentlemen, I'm welcoming you on the second day of our conference about post-mining forest and landscape restoration. I'm happy we join in quite a high number, and we will continue the similar manner like yesterday. So you might see the program before. We have still one speaker missing, but maybe will appear during the session. We will actually ask individual speaker to share screen from their own computer. This can be done by green button on the bottom of your screen. So basically when you scroll your mouse to the bottom of your screen, there's usually toolbar appear. In the middle of this toolbar, there is a green button which arrow up. And if you click on it, you can share your screen. And then you can, drive this yourself. You can change the slides yourself and so on, according to your convenience. I will ask Dr. Hedinec to chair this session, and with that, I'm going to, wish you enjoy the meeting, and yeah, maybe I will finish some of this housekeeping information. So basically, this is 10 minutes for the speech and 5 minutes for discussion. And after each session, there is a coffee break. We do not provide coffee, unfortunately, because we are technically not able to serve coffee from Sydney, Australia to Knoxville, Tennessee, where our participants are coming from. So you have to unfortunately serve your coffee yourself. But at least to do something, we organized a breakout room during each coffee break. And in this breakout room, the people who have speech in previous session will be automatically associated with, so they will appear in the breakout room. And for each of other users in the toolbar, they will appear the tool to go to breakout room. If you click on it, you see a list of breakout rooms, and you can see who is inside. And you can join the breakout room, which, individual speakers, or you can even make appointment with some other and meet with other people with a free breakout room and have more private conversation face-to-face as much as is possible in this online environment. So with that housekeeping information, and then certainly the session is recorded and is streamed online in the YouTube. So that was before the YouTube link. So if some of your students are interested, or if you, for any reason like the time shift, you miss some of the part of the session, you might actually see it again on YouTube. So with that, I would hand the words to Dr. Hedinec and ask him to start today's session.
So thank you, Professor Froese, for a nice introduction and for welcome speech. And good morning for everyone. And I have great pleasure to invite first speaker, who is Xia Kang. So please, you can start now. Please unmute first.
Share your screen if you
And
can.
Can you hear me?
yes,
Hello, everyone.
Yes, we can.
Can I just start? It's just 9:05. Is that okay?
You can start. You can start.
You can
Okay,
start.
right.
Can you share your screen?
Okay. Right.
Yes.
Excellent. Can you put it to full screen mode? Great.
Sorry, maybe I can put the pictures of your, hide this picture. Okay,
This is good.
I can see.
We can see everything on the screen.
Great. I am now I'm going to talk about the changes of root microbial populations of natively grown, and this plant is called commonly known as yellow bluestem. It's a perennial grass, and another one is narrow leaf cattail. It's another water plant. It's normally grown in the water. Those changes of the microbial populations during the natural attenuation of vanadium titanium magnetite tailings. I am just now affiliated with the Chengdu Institute of Biology, and all this project is done by my boss in my previous studies. So let me first introduce the study area, and I will give you some background information about the study area. It's vanadium titanium magnetite mine tailings in the central southwest part of China. It's on the borders of Sichuan province and the Yunnan province. It's around a small city called Panzhihua City. It's a region noted for intensive mining activities over 14 years. And all over these years, there has been heavy industries, and there are lots of mine tailings as a result of the mining activities. And in this region, there is it boasts of Asia's largest VT magnetite mine tailings and its mine sites. So it caused a lot of pollutions and great health concerns. So this is the left figure is the actual picture taken on the site of the mine tailings. It's basically an embankment. It's a very big, huge dam with millions of tons of magnetite tailings, and they are left there for a natural attenuation process. And all over the years, there has been a lot of natively grown plants which are growing all over the place on the tailings. And here you can see on the right-hand side, this is the cattail, and there is the blue stem, which are naturally grown. It's not grown by humans. It's just a natural occurrence. So let me. Can you see the laser? The laser pointer?
Yes, I can see the laser.
Okay,
pointer.
right. This is better. This is the cattail plant, and this is the blue stem. They are mixed together and growing in the mine tailings. And the black stuff is the tailing soil. It's a mixture of natural soil and mine tailings. It's rich in very high concentrations of heavy metals, especially iron and titanium and vanadium and some others. So let me do the next slide. So the material and the methods. So the soil samples were collected and transported to the lab for analysis. Here we chose the two plants which are dominant species, the blue stem and cattail, as previously described. And we took the bulk soil of the blue stem and the rhizosphere soil of the blue stem, and also the bulk soil of cattail and the rhizosphere soil of the cattail plant. And the plant-free tailing soil served as the control. And for the soil analysis, we did firstly the physical chemical properties of the soil itself, such as available nitrogen, phosphorus and potassium, pH and TOC, and the microbial biomass carbon nitrogen, and some soil enzyme activities, such as nitrogenase, catalase, invertase, and phosphatase and urease. And also we did the measurement of the heavy metal contents. And there's also the soil microbial communities analysis, which included the relative abundance, which is expressed as bar plot and heat map, and the discriminant analysis of the microbial populations and alpha diversity, and also, as always, beta diversity, such as NMDS analysis and Permanova. And also we did some correlation analysis to find out the relationships between the environmental factors and the microbe populations. And we did CAP analysis and mental test and co-occurrence network analysis. And finally, we did the microbial functional predictions to see which functions are present in the soil. So, and also we did the plant analysis. We collected the plant samples and bring them to lab to test for the heavy metal contents such as cadmium, chromium, copper, etc. And all these metals are present in the mine tailings. And we also did calculated the biological accumulation factor, or BAF, and also translocation factor, and the geoaccumulation index to assess which metal is best absorbed by the plants and whether the plants have the ability to remove them as a means of phytoremediation. So next slide. First, the results of the mine tailings and the metal contents in the mine tailings soil. And here we can see the available iron. All these are the plant associated soil samples. They are very low and they are drastically reduced compared to the control. This is the control. And this means that the plant might have some abilities to absorb the iron or transform them into some other insoluble states. And and also the available nitrogen was increased compared to the control. And this result concludes that the plants might have the ability to alleviate the soil, reduce the soil stress and increase the nitrogen availability in the soil. And this might be associating with some nitrogen associated bacteria or microbes. And also we see the microbial biomass carbon was also significantly increased compared to the control. And all this happened in the rhizosphere soil of cattail and in both the bulk soil and the rhizosphere soil of the grass. So we further did some analysis on the accumulation of these plants. And here we found that lead was accumulated by this plant. This is the cattail, and magnesium was accumulated by also by this plant, but all the other metals are not accumulated. So these two plants, the cattail and the blue stem, are not accumulator of all these plants except for lead and magnesium. And here is the profile of the soil pollution. Here you can see cadmium caused severe, extremely high pollution level to the soil, but we didn't find it in the plant. Maybe it's not absorbed by the plants or by some other interference. We didn't detect them. So, and here you can see chromium caused strong pollution to the soil, and copper and iron caused moderately pollution to the soil. And although lead was not very pollution, not much of a pollution to the soil, but it's effectively absorbed by the plant. So here we did the microbial analysis, and which revealed that at the class level, alpha proteobacteria was increased. Here you can see in all the soils of the plants compared to the control. And interestingly, gamma proteobacteria was reduced, or significantly reduced compared to the control. And we did some analysis on the at the general level, and which we found that out of the 16 most abundant genera, 12 were increased, 4 were decreased, and all these increases are very significant. Here you can see this is a top 16 genera, and the Rhizobia, which controls the nitrogen fixation function, was dramatically increased, and also this Massilia was also significantly increased. But this one, Acinetobacter, was significantly decreased or reduced, and also Theobacter bacillus was also significantly reduced. So this is just an overview of the microbial taxa found in the soil. And here we did the LDA analysis. The discriminant analysis found that in the control soil, Acinetobacteria was the predominant species, the taxa, and among all the taxonomic levels, alpha proteobacteria was the most prominent in the rhizosphere soil of cattail, and Proteobacteria was the most prominent taxon in the bulk soil of blue stem. And at genus level, it's a bit different. Ophidius, this genus was the most significant species in the bulk soil of blue stem, but Massilia and Rhizobia were the two most significant species found in the rhizosphere soil of cattail. So this results says that there are differences, there are different preferences of these plants to choose their preferred microbial populations. And we did the analysis of the alpha diversity. And here we found the Shannon index, which shows that all these figures are very high compared to the control. After being grown with the plants, the microbial activities, the microbial populations are increased compared to the control. And this is obvious. And here on the right-hand side, this is the NMDS analysis revealing the beta diversity. Here we can see the stress level is 0.084, which means this is a good one. And this figure indicates that all these differences are shown on the map, which is very big. The plant-associated soil samples are quite different from the plant-free tailings sample. And this one is the analysis of the association or the relationships between the soil factors with the microbial populations. And here we found that available iron and TOC has the longest arrow, which means that they have a higher control over the microbial changes. And the mental test shows that in the plant of blue stem, the correlation was most significant with many different soil parameters, such as the metals and the microbial nitrogen, and also some interestingly, all these enzyme activities are closely associated with cattail plants. And here is the network analysis. We did this to review the co-occurrence network of these microbial populations in these two different plant species. In the blue stem, it seems that it's not very complicated, and this is the parameter of the network graph. The total triangle indicates that the higher total triangles means there are more complicated networks. And this is obvious that cattail harbors are more complicated, sophisticated microbial communities compared to the blue stem. And finally, we did the functional prediction. And the overview of this heat map shows that the yellow-blue stem preferred nitrate and reduction functions, while the narrow cattail preferred nitrogen fixing functions, which are quite different. Although they live in the same grown, in the same maintaining stem, they choose to absorb or attract different microbial communities. And this is the result of my project. And so thank you very much for your attention. Can I stop sharing the screen?
Thank you for a very nice presentation. And now we have time for 3 questions.
Yeah, do you have any questions? Please
Yes,
just
we have.
ask.
Yes.
May
Okay.
I ask? I have basically 2 questions which are part related.
Yeah.
You mentioned the cattail is actually focused on a nitrogen nitrate reduction. So where is the source of nitrogen in that environment?
Because we did some other previous researches, we found that we identified an isolated rhizobia. We used the culture-dependent method in our previous researches. There are actually rhizobia living in the mine tailings, because there are Lucella, the Cecophana, the legumes that are found also around the tailings pond. Okay. So we think that might be some.
They use nitrogen produced by another plants.
Yeah. They might be associated with nitrogen fixers.
I have a question to your network analysis, because many people infer some parameters of network, like network stability, from the network analysis. So can you do similar conclusion that some of the networks you observe show higher network stability than the others?
Network analysis is quite tricky. It's just to review the relationships, the correlations, but it doesn't mean causation. It doesn't mean the actual, which is the cause, which is the reason. So there are some parameters. We can only infer from the parameters that which is more complicated and which is simpler. So the complicated one means that there are more different associations of microbial populations, and the simpler one means it's just simple. And another thing is that we can infer from the network that which is the keystone species, which we did, but we are not sure because this is purely based on the mathematical calculations. Some other papers say that which one has the highest nodes or some connections, which this one can be classified as the keystone taxa, but it's not universally accepted. So it's quite tricky.
Mm-hmm. So second questions, Prof. Michal Bonkovsky. You can.
Yeah, I have a question and a comment. So one maybe the comment first. So the rhizobia associated with grasses, they are not nitrogen fixers. So rhizobia are common endophytes also in grasses and cereals, but those are not, they do not have the ability to fix nitrogen. It's only the rhizobia associated with legumes.
Yes, legumes. Sorry, I correct. I was just going to mean legumes. There
but it is very common. So all the microbes you showed, they are very familiar to me. Marsilia opitutus and so on. They are very common rhizosphere taxa. And so in your NMDS, you showed the VT. That was the control. So that was the main mine tailing.
Yes.
And the bulk soil was, you sampled, that was close to the plants?
Yeah. The bulk soil is close, but not in the rhizosphere of the plant.
Okay, so that means that the bulk soil, so where the plants grow, there the soil conditions apparently are quite different from the main mine tailing still.
Yeah.
We had no overlap there. Okay, so the plants choose some sites where they can still grow, and with the microbial community, you can really detect these differences.
Yeah.
Okay, thank you
very much. Thank you.
Any other questions, please? If not,
I have chicken or egg question. So do you think the reason why the bulk soil is different is because the plant chose a different microhabitat, or that the plant effect penetrates so large around the plants, so they actually affect not only rhizosphere but also surrounding bulk soil?
Yeah, I think they will.
I mean, did you do some experiment like you will plant the plants into the plant-free soil and look if they are actually change the bulk soil? Or
We haven't done that yet. This is just the on-site study or survey of the populations of the microbials.
So thank you, Xia Kang, for very nice presentation. And now I am happy to introduce another speaker, who is Lukáš Jačka. So please, floor is yours.
Thank you. Thank you very much for your invitation.
Yes, okay. Now we see.
Can you see the screen well, please?
Yes, we can see.
Okay, well. Good morning, ladies and gentlemen. Thank you all for joining the presentation. My name is Lukáš Jačka. I work for the Czech University of Life Sciences, and presented research was done together with Professor Jan Froos from Charles University and Dr. Alena Wolmsley from Czech University of Life Sciences. Now I am going to talk to you about the effects of different tree species on infiltration and preferential flow at reclaimed soil, which is developing after lignite mining. Presentation is divided into the 5 parts. On the left side of the slide, you will be able to see highlighted actual part of the presentation. So let's move to the first part of the presentation, which is motivation of the research. Here you can clearly see that different tree species can affect soil developing strongly differently, and it's really nice how different soil profile can be created under different tree species. On the left, here is example of alder tree, on the in the center oak tree, and on the right spruce. Restoration of hydrological functions is crucial for the whole ecosystem recovery, and our research is concentrated to the important part of the hydrological cycle, which is infiltration and preferential flow. Preferential flow and infiltration are little studied, and our research question is: what is the role of the litter quality and root system of different tree species on these important hydrological processes? Because infiltration is only the one process which enrich soil profile by water. The major hypothesis of our research is that under tree species characterized by more easily decomposable litter and lower CN ratio will exhibit higher Ks value infiltration and cross-section size of preferential flow. Aims of the research are to explore differences in hydraulic conductivity and related soil properties like pore size distribution, earthworm abundance in topsoil and subsoil, and to examine a preferential flow in soil profile using dye tracer tests, and to analyze dye stained patches and role of biostructures, namely root created structures and worm created structures on preferential flow. 3 different soil were tested, and important information is that we selected different monoculture of alder, oak, and spruce in clay spoil heap. The study was performed in Vintířovská spoil heap, which is part of the Velká Podkrušnohorská spoil heap in northwest Czechia. It's in the Central Europe, close to the border with Germany. And we benefit here from the existence of something what can be called common garden experiment, which means planting of several tree species in small monoculture patches. Here you can see picture of the patches in the Vintířovská spoil heap, and we selected 3 study sites. These study sites were directly close together and were dominated by 3 different species, alder, oak, and spruce, and the substrate for these sites were the same, and the edge of the forest was also the same. So 3 species factor should be the only one which affects soil development. We performed here you can see detail of the positions of samplings of KS tests and dye tracer tests and others. Let's move to the methodology. All hydrophysical measurements were performed in organic rich topsoil and mineral subsoil. Field work was restricted to 2 consecutive days to eliminate effects of temporal variation on soil moisture changes and other soil physical changes. And we used standard method for KS measurement, Guelph thermometer, and undisturbed soil samples of volume 100 cubic centimeters to estimate pore size distribution from soil water retention curve measurement. We also excavated soil cores for determination of earthworm population, which were extracted by hand sorting and determined it down to species level. We also made dye tracer infiltration tests with brilliant blue water solution of 5 grams per liter. Here are details about the methodology, and we also analyzed dye patterns in soil profile in macro scale and micro scale. We analyzed the role of cracks, root, and channels in 2 scales, and also worm-created structures. Detail about modifier method are mentioned in the paper mentioned here, and let's move to the main part, results and discussion. According to our hypothesis, the highest macroporosity, the highest number of earthworms, and the highest value of key infiltration parameter, which is saturated hydraulic conductivity, was observed for alder stand, and the lowest value for the spruce. Alder stand had the lowest CN ratio and the most easier decomposable litter, and so this was in agreement with our hypothesis. What should be mentioned also is that total porosity was practically the same for all tested three species. So it means that pore size distribution and its connectivity affects differences in infiltration and preferential flow. Here you can see dye tracer distribution patches on vertical cross sections through the soil profile, which were made in the center of the infiltration area, and you can clearly see that distribution of water was very different among the tested 3 species. In the topsoil of older soil, you can see high lateral flow, which is caused by a layer of aggregates which are connected to the dryosphere and activity of earthworms. In the center, you can see distinct effect of the root system of oak, channels around this a strong system of roots cause distinct macropore flow, and you can see dark blue area here, which is one good example. And lowest preferential flow was observed for spruce forest stand. Here you can see these findings in detail because the lowest infiltration rate was observed, the highest infiltration rate was observed for alder tree and the lowest for spruce. And here you can see what is connected to high infiltration rate. This is high lateral preferential flow in topsoil. Here you can also see the layer of aggregates which were formed in the dryosphere in the topsoil. And here you can see also the abundance of earthworms. And I would like to say that I think that probably the role of epigeik earthworms for forming the dryosphere layer of aggregates was important for preferential flow in topsoil for alder stand, and fermentation layer was very important for water flow fields in spruce stands. Here you can see different water distribution patterns. On the left side, you can see dryosphere of alder tree, where is light blue. It means that water went quickly through this layer of aggregates in the dryosphere, and on the right side you can see fermentation layer of spruce, where water was absorbed and flow very slowly because of the complicated pathways in the fermentation layer. And here below on the left you can see role of the biostructures in mineral subsoil of alder, where is a burrow of anessic earthworm, and on the right side you can see anessic earthworms in oak. In spruce, anessic earthworms were not observed. Also, epigeik were not observed. Here, I would like to say that dyed cracks in the soil profile were surprisingly there were lots of dye cracks in the soil profiles, but they can be connected to the rhizosphere and dryosphere, which is basically shown in this table in macro scale. There is high amount of cracks in alder, and if we look in the micro scale, we can see that many of these cracks can be are connected to dryosphere or root. It also has to be said that most of the dryosphere and rhizosphere is connected together by water infiltration pathways, and it can be seen, for example, here in root microscale thin soil section, where 59% of the dyed soil can be traced to the dryosphere, and similar situation is also for oak. Here you can see it. So Let's summarize the effect of three species on the cell recovery. Three species affect infiltration and preferential flow in soil profile by their specific root systems, and to a large extent indirectly by effect of litter quality on earthworm population, which then enhance macroporosity and its connectivity. Here you can see some example where the size of the preferential flow, infiltration, and KS is highest for all the three with the easiest decomposable litter and with the lowest CN ratio. Here you can see example of roots in oak system, and fermentation layer of spruce block water flow in the soil profile partially. Therefore, the size of preferential flow is the lowest. So let me conclude. We observed different preferential flow and infiltration parameters for the tested three species. The important message is that all hydrological processes can be distinctly affected by three species planted, and that preferential flow is playing dominant role in spoil heap, and soil biostructures play important role. Earthworms contribute to the porosity around the roots and open blinded cracks in clay stones and form layer of aggregates in topsoil. Open blinding cracks in clay stones can then store water for the period of drought, and that is one important finding, that it's working like that probably. Future challenges are that research should be expanded to other tree species and mixed stands that are desirable from the ecological point of view. And that's all from me. Thank you very much for your attention.
So thank you, Lukáš, for a very nice presentation. Now we have time for one question, please. Somebody have? So
maybe the role of these endogenic earthrooms, I found it very interesting. The epigenic earthrooms.
Epigenic, okay.
Really interesting.
Okay, I was also surprised, because I think that sometimes they are in the literature underestimated in comparison with anesthetic earthworms. And here you can clearly see, I was really surprised when we excavated cross sections, how important are aggregates in the topsoil for hydrological behavior.
Thank you.
You're welcome.
Any other questions?
I might have a comment to that. So what's really neat on that study is actually show that not only the actual pores created by worms, but also fact that they assure connectivity of the cracks and pores, which was already there, but it wasn't connected.
Yes.
It's really important for general infiltration and water storage.
Yeah.
Any other questions or comments? If not, now it's my greatest pleasure to introduce another speaker, who is Martin Bartuška from SOVA Research Infrastructure, and Martin will talk about usage of chronosequences methods on post-mining sites. So Martin, floor is yours.
Yeah, hi. Nice to meet you. I'm happy to see some of my friends I haven't seen quite a long time. I will try to start my presentation and share it, because this Zoom is new for me. I would say Zoom. Sorry for that. Share screen. Yeah. Here. This is if I don't go here. So No, this is different.
Share your screen. It's a yellow.
Yeah, but
it's a yellow button on the.
You should have opened it, and then you should have opened the file, yeah, first, and then in the middle of your screen there is a green button. There's an arrow up, say share screen. So if you click on it, you should get choices of files you can share.
Yeah, but
your file should be open first, yeah.
Yeah. Yeah, it looks like, yeah, I thought, yeah. So this, open, yeah, yeah. Sorry for that, but I can't get.
Tam se napřed musí kliknout na ten obrázek a potom kliknout na sdílet nebo share teprve, jo?
A tu vybrat, co budete dělat celá obrazovka, sdílet.
A potom kliknout vpravo sdílet.
Or do we have your presentation?
No, I haven't sent the presentation. I didn't send it. Yeah, sorry.
I see something.
Okay, yeah, so.
It seems to coming.
Yes.
Should be okay now.
Ještě prezentační mod.
Yeah, so.
Vpravo dole takové plátno.
Yeah, it looks.
Or F5, sometimes it works.
I can see it on my, yes, I'm sharing it. Is it sharing now?
I can see just the basic mode.
It is, yeah. And if you go to presentation now and click on the full screen. Okay, leave it like it is. I think we can see it. So better than nothing.
Yeah, sorry for that. Sorry for that. So when I thought about my presentation, I decided to make some conclusions about what my work was done and what was the most important thing about this research I did. And what I found out, it's the uniqueness of this environment. It's the known history of the soil pathogenesis, the history of the ecosystem there, the way we are reclaiming it or unreclaiming it, and there are possibilities to find repeated patterns in different ages or different stages of their development. So that's the cross-sequence method. One of my research was about repeated measurements of cross-sequence after 11 years. There was done research by Ms. Shorkova, and she did measurement of all these cross-sequences. And after 11 years, we decided to remake this research, and this was the most interesting and nice way to mitigate the variations of the size or the basic variations which could occur in the substrate, especially with carbon in some post-binding sites, when you can have a little bit more of or some pieces of small carbon or some coal, which can changes your results. With this combination, we could make some real-time real-time estimation and changes. Please, what?
I just see only first slide. I'm sorry. I just
yeah. I am not moving right now. I'm just trying to show.
Okay,
so
perfect. I'm sorry for disturbing you.
Yeah, it's okay. So what we were looking at first, we were looking for the pH. So we were the values.
Can you move page down to next slide, maybe?
I'm going down. Is it visible? pH now?
No.
No. We still see just the
Yeah,
first.
now, yes,
now it's pH. Yeah.
Yeah. And now it's moving.
Yes.
Yeah. Now it's moving.
Yeah. So when I have it in as a whole window, it doesn't work. So yeah. So I will leave it this. So as you can see, there was 11 sites which were measured, and after 11 years we were able to see the changes. So so when the value from 11 years and now, and we can divide it in 11 years, we can see just a new change. We can see here in the chart the lower one. So here it's visible the changes that at the beginning for the young sites we can see some changes. It's pH, which is slowly going down, and with age you can see that there is some changes. There is an increase. Because our substrate is alkalinic, so we can say that when the substrate is alkalinic, at the beginning of the sputogenesis, the pH is decreasing, and on the places where was pH lower, it was increasing. But here there is a visible nice way how it's changed during these 50 years and how the annual rate is how big is the annual rate. We made these with carbon as well because it's carbon, one of the main things we are looking for. So we saw that the carbon stock with age is increasing, but the speed of increase, the annual increase, is the fastest in early stages of 10, 12, maybe 30 years. But with the age of the sites, we can see that the annual increase is going down. The content of carbon is rising with the age of these sites, which everyone knows. But if we can, for the dynamics of it, that's the annual content increase, we can see the higher increase is about 20, is between 10, 20, 25, up to 30 years. And then the increase is going down, it's slowing, and at the high age of these places, it's almost at 0 or 0. Yeah, it's good. It's going down. The same pattern works for nitrogen as well. Yeah, the initial increase in 10, 20, 30 years, and then decrease of the speed of the content increase. The different pattern we can see on phosphorus, because phosphorus is just in soil, so there is no way to incorporate a new one. And here is visible that the lowest amount or the increase of phosphorus, the lowest about 20, 30 years, and that's because it looks like the phosphorus is going to be less accessible because it's incorporated in organic or on the body of the trees, on the body of the herbs or grasses. So maybe there should be there starts to be some limitation of phosphorus for the for carbon and nitrogen, and phosphorus starts to be limiting for the ecosystem development. As a conclusion, we can say that accumulation rates decrease with the age of plants, and this supports the soil carbon saturation theory, and the carbon and nitrogen have similar trends. The content of phosphorus is decreasing with age because it's incorporated in bodies of plants. What we look then, sorry, it's in Czech, I didn't realize it. Something we wanted to see the differences in fractions of the carbon in soil. Because when we were looking just for the carbon, it doesn't say a lot much about how the carbon works in the soil. We made basic fractionation, density fractionation for light friction. This is LF, this is the free one, here the biggest one on the macro aggregates or between macro aggregates, and bounded friction, which is in macro aggregates or in micro aggregate. We wanted to see the differences. We wanted to see the differences in the distribution of carbon for unreclaimed and reclaimed sites in age of 10, 20, 30, again, some in these chronosequence, which is the connecting bridge for all these studies. As we saw, the unreclaimed places has lower content or lower amount of light friction, even the bounded friction. The reclaimed places where it's elder is really has let's say 10 times more of this carbon than the unreclaimed in both fractions. Yeah, I want to show this first. When we make the when we look for fungal bacterial ratio of these fractions, of these bounded and light fractions for middle age and late age, again middle age and late age, we saw that the fungal bacterial ratio is lower for reclaimed places, so that means there is more bacterial, and for unreclaimed there is more fungi there. Using PCA test, it showed us 69 of the variabilities, and the strongest one was by reclaimed and unreclaimed axes, because reclaimed and unreclaimed, and the second one was 18%, and it was the age. So what we can say that the age isn't such interesting, or no, the age is not the main thing of the biology structure we were looking for. It was the amount of light friction and bounded friction, and the light friction is more connected with unreclaimed places. It's the not incorporated matter, and bounded fractions is connected with reclaimed sites, which is connected with micro aggregates. When we took some cuts from the soil, so this is the first initial stage of the soil, and then you can see how it moves forward. On the top, this is a reclaimed soil cut. Under there is unreclaimed soil cut, and under the microscope, we can see that reclaimed sites has big bioturbations by worms. So the organic matter is incorporated in the soil and is connected and bounded in macro aggregate and micro aggregates. And the unreclaimed sites, there is no bioturbation. There is more some Orebatida or isopods which are feeding the litter laying on the surface, and this later makes something, let's say, like sieve, which doesn't let the, let's say, some cast or go through to the surface or to the soil, so it's not incorporated in. So presence of worms and incorporation also organic matter influence for properties. Differentiation of carbons in fractions is influenced by macrofauna, and microbial communities are influenced more by type of fractions than by time. Microbial communities does not change in times, but abundance is changing. All these things were easily to research because of the chronosequence and because of the uniqueness of this environment of post-mining sites, which should be thinking if someone is working on post-mining sites. I think it's interesting to try to find this way, this chronosequence and use it, because the data are there much more solid, and you get rid of variation of some possibilities in states. So thank you for your attention, and I'm here for your questions.
So we are slightly delayed, so I will move questions to the discussion part.
Yeah.
But thank you for presentation. And
you will join the breakout room, and the people can ask you the question.
Okay. And now it's my pleasure to invite another speaker, who is Sangeeta Mukhopadhyay, and she will talk about evaluation of reclamation success of coal mining sites through the soil quality index. So please, you can start.
Yes.
Now we see your presentation. Please.
Yes.
Okay.
It's visible. Okay.
Yeah, it's visible now. Okay, perfect.
Okay. A very good morning to one and all. First, I would like to thank Professor Firoz and his team for giving me this platform to share my research work during this COVID time. Now, myself, Dr. Sangeeta Mukhopadhyay, actually I am carrying out research in mine soil reclamation for about a decade, and associated with reputed soil scientists like Dr. R.E. Masto from Central Institute of Mining and Fuel Research, Dhanbad, and Professor Maiti from Indian School of Mines, Dhanbad. Now, yesterday in the first session, the mining scenario in India has been vividly described, and was highlighted that the overburdened dump material left post-mining are biologically inactive and lack plant nutrients. So scientific reclamation of these derelict sites is a challenging task. And in India, forestry reclamation approach is generally followed for restoration of these derelict sites, and by the plantation of tree species. Now, what is important here is, in the management perspective, mine owners, stakeholders, they always desire to know the health of reclaimed area land and the time required for the development of a self-sustaining forest cover. That is the status of reclamation. So soil quality assessment of chronosequence sites is important to estimate the reclamation time. And this progress of ecosystem recovery can be determined by identifying soil quality indicator parameters. And based on these soil quality indicator parameters, an integrated soil quality index can be developed to ascertain the stage of ecosystem recovery. The higher the index score, the better will be the soil development. So the objective of the study is to identify mine soil indicator parameters, and then integration of these indicator parameters to develop the index, and to evaluate the success of the proposed index, and to develop tools for assessment of reclamation success. The study was conducted in the reclaimed chronosequence sites at North Karanpura area, Ranchi. Now, as we know, as open cast mining goes on, it can continuously creates new sites over an extended period of time. And as these sites are revegetated at different time periods, similar mine sites with various ages of plants are available to us at one time. So this gives an unique opportunity to study the progressive development of soil development in post-mining sites. It's an approach that helps in assessing the rate and direction of change in various petrological processes. Now, these are the different aged reclaimed dumps, 2 years, 5 years, 7 years, 17 years old dump that we have selected for the study. And as for mine soil quality, we don't have any standards like air quality, water quality. We compare with the nearby, compare the data with the nearby sal forest, that is the control site, and also with the fresh mine soil. These are the tree species growing in these reclaimed sites. And we found that the 17 years old site was dominated mainly by Acacia siamia. In rest of the tree species, the Albertia shishu was dominating, relative density was high, DVH, height, canopy cover, all these parameters were found to increase with age of reclamation. Now, these are the very fundamental soil parameters, physical, chemical, and biological parameters that has been shown over here. Was analysis was carried on, and here in physical parameters like coarse fraction, it is actually a very important indicator mine soil parameter, because if coarse fraction is high, greater than 50% in mine soil, it will inhibit root proliferation and nutrient availability to the plants. And so here coarse fraction we found was very high in the fresh mine soil compared to that of the reclaimed sites, and sand quantity percentage was also higher in fresh mine soil, where silt and clay was higher in the reclaimed sites. Bulk density was higher in the recently reclaimed sites compared to the older sites, and bulk density difference may be also attributed to different mining equipments used and organic inputs in older sites. And few chemical parameters like soil pH here in mine soil was acidic and no significant trend was obtained. And this mine soil pH also depends on the overburden, parent overburden material, and soil organic carbon increased with age of reclamation due to accumulation of organic matter in the older sites, litter, deep litter, decomposition, carbon mineralization taking place. Available nitrogen and phosphorus also followed the same trend, and carbon cation exchange capacity, which is also a very important parameter for it determines the soil fertility and nutrient retention capacity. In fresh ovidum, due to its porous structure and high leaching, the cation exchange capacity was less, and as the dumps got stabilized, it increased with age of reclamation. With increasing clay fraction, the base saturation was also highly less in the recently reclaimed dumps and increased with age of reclamation. Now, coming to the few biological parameters, soil dehydrogenase activity is an important endoenzyme and catalyzes several biochemical reactions we have shown here. Its activity increased with age of reclamation due to higher vegetation cover and organic matter input in older sites. And microbial biomass carbon also followed the same trend. However, we find that the 17 years old dump is less than that of the 5 years, because here it was improvised condition and yes, ground vegetation was very sparse and low tree density was found in the 7 years old site. Average soil CO2 fast was highest in the 17 years old site due to high accumulation of organic matter and greater microbial activity than the younger dumps. Now, here I have actually shown few data, important soil physicochemical parameters considered for developing the index. However, later we thought that holistic assessment of mine soil quality should include not only these physical, chemical, and biological parameters, but as well as the presence of hazardous and potentially toxic elements. Actually, coal mine soil also contains pollutants like polyaromatic hydrocarbons PAH and trace elements inherent in coal. So later, a holistic approach index was developed, taking into account as much as possible all the physicochemical and biological parameters, available plant nutrients, 16 priority POH, PAH, and trace elements. And we concluded that the soil parameters, all this role of all these soil parameters in the role in the may influence soil development, and it was published in this paper. Now, how to develop this index? So in this paper, we considered about 50 parameters. Now, suppose we are going for assessment of rhizosphere soil quality or chronosequence site. Once we develop an index for a site, we can apply that indicators for assessing the other sites. We don't have to analyze all those 50 parameters again and again. So this is one utility of this. Indexing involves three main aspects actually. Choosing appropriate indicators for a minimum data set, transforming the indicators to scores, and combining the indicators into an index. Now, here I will show you this is the equation based on PCA. Here it is the summation of the weights and the scores. Weight is the principal component weighing factor, and S is the score of each variable. Here, the score has been calculated based on this equation. And all this data for parameters, physical, chemical, and soil parameters were subjected to PCA to identify the key indicator parameters. Now, the first three pieces with eigenvalues greater than 1 was taken. Under a particular principal component, PC1, only the variables with high factor loadings were considered and retained in the index. Here, in principal component 1, we find organic carbon, CO2 flux, and dehydrogenous activity having greater weight. Now, the method is we select these three variables with highest factor loading and correlation is done. And if they are well correlated, then the variable with the highest factor loading are chosen for the index. And if not correlated, then each are considered important and retained in the index. However, here, the one with the highest score is soil organic carbon. We did not find, we find it not be wise to depend heavily on this. So we considered the rest two and retained in the index. In the other PC2, both these parameters, soil coarse fraction and moisture content, was not correlated and were retained as indicators. And in PC3, the base saturation was the last parameter. So these are the indicator parameters selected based on this principal component analysis. And now we know the score and the indicators. Now, weights of the selected variables were determined by percent variation in the data set. Within a particular PC, the weight was distributed among the correlated variables as per the factor loading. And if not correlated, the full weightage was given to them. Now, this figure shows the principal component, this index score, and it also presents the contribution of each parameter on the calculated index value. Lowest MSQI was found, this index score was found in the 2 years old down due to lesser score of all these 6 parameters, while higher value was obtained in the 17 years old down. Here we have shown the values, and now, this index we have developed need to be validated, otherwise of no use. Now, this index was validated with plant growth parameters like endpoint variables like tree height, canopy cover, DVH, and it was found to be well correlated. Now, this index recorded for was higher for the 17 years old site, which shows that it's close to recovery of the near to the natural forest. So we recommend that eco-restored mined soil sites with this index score greater than 0.5 could be considered as ecologically sustainable or reclamation is satisfactory. In other words, at least 50% of the soil functions has been restored in the sites. Now, this index can be employed as a tool for evaluation of the reclamation success. And we can apply this for the other mining sites, though we recommend a proper validation of the index. Now, besides assessment of this ecological, besides assessment of the reclamation success in the mine soil by this indexing method, this method can also be employed for the selection of different tree species suitable for reclaiming the mine sites. And also this study, we have published similar study in this journal. And we have developed the holistic approach, this index for coal mine soil, considering almost about 50 parameters, and similar studies can be also done for selection of suitable amendment. Presently, I am working on the development of low-cost, slow-release composite biochar fertilizer for mine soil reclamation. And that's all, I think. Thank you.
Thank you for nice presentation.
Thank you, sir.
Now it's time for one question, please.
I have one question. What is added value of this index? You show by quite complicated way it nicely correlated with the basic vegetation parameters. But my question is, it is really easier to measure all that 5 soil parameters like, you know, enzyme, respiration, soil properties, than to see how the plants perform? To my opinion, you know, to measure DBH, tree age, tree size, and tree density would be much easier than measure all the soil parameter you consider.
Sir, actually, we are considering a lot number of parameters for few sites, and then coming to a less number of parameters, we are deriving the indicators, and those parameters we will measure for the rest of the sites. For the rest of the sites, we won't go for all the parameters. Like we have taken into consideration heavy metals, plant nutrients, everything. But what are the key parameters that we will give press and that we will analyze for the rest of the parameters for different reclamation sites we have done. And this is based on
choose was quite comprehensive, and it still seems to be much more difficult to obtain than the plant parameters.
No, sir, we have analyzed the vegetation parameters, DVH, height, and all those. That we need to do for all the plants.
So if you have that, what is the added value of the index?
By indexing, we can come to the conclusion which is better by some statistical tool. We are applying different statistical tools that we can tell concretely, come to a conclusion that this 7 years old dump has been better than this one because.
But that's basically circular evidence, yeah? Because you see it from vegetation, and then you prove also that soil is better because support better vegetation.
Yes, yes, sir.
Okay, we can discuss more in
Yes,
breakout
sir. Thank you very
sessions.
much.
And now I would like welcome the last speaker, who is Sneha Bandyopadhyay, and she will talk us something about comprehensive soil quality index and its usage in reclamation of coal mining degraded lands. So please, floor is yours. Okay, it's no problem. Please unmute. Please unmute you. Yeah,
is it visible, sir?
Yeah, it's visible and we can hear you. Yeah.
Okay, okay, sir. Thank you. Good morning to all respected delegates present over here. I'm Sneha Bandyopadhyay, going to present my work on development of comprehensive soil quality index for suitability ranking of plant species used for reclamations in coal mine degraded land. Presently, I'm pursuing my PhD from IIT ISM Dhanbad, India, under the respected Professor Subodh Kumar Maiti. And obviously I would like to thanks to all the respected organizer for giving me the opportunity to present my work over this platform. So slide is not proceeded. So, my presentation synopsis mainly includes the overview of coal mining and its related socio-economic development, as well as the land degradation related to it, and the reclamation process, which converts that mine spoil, the generated mine spoil to soil, then hypothesis and objectives, my research activities, and then the results and summarizations. So since the industrial revolution, mining in industries afford most of the indigenous energies like fossil fuels, spatially, that we rely for the industrializations and the energies production. Amongst the conventional fossil fuel like coal, oil, and natural gases, coal is the second largest source of primary energies and that accounts for 30% of total energy consumptions globally. As of 2018, the world recoverable coal reserves were estimated about 1,055 billion tons, out of which about 75% are located in most of the 5 countries like USA, Russia, Australia, China, and India. So being the dominant source of global energy arena, coal fulfills about 38% of global electricity demand and 84% in India. That is the world's third largest energy consumer after China and USA. So but the problem is the majority of the Indian coal reserves found in the depth up to 300-meter and usually excavated and obviously under the forest cover. Maximum are under the forest cover and are usually excavated using the surface mining techniques more than 90% cases that alters the natural landscape into degraded environment. So surface mining mainly removes the total vegetations and topsoil cover, excavated and dumped the rock overburden, that is the overburden material, OB material, initially outside the dumps, that is the external dumps, and afterward used for backfilling of mine voids like internal dumps. So these OB dumps are dominated by sedimentary rock debris and may pollute the environment if not properly managed. So moreover, deforestation due to this mining activities significantly affect the hydrological regime, the earth surfaces, decreases the carbon stock and the soil fertility, as well as destroys the ecosystem carbon sink that causes the major depletion in biodiversity as well as global climate crisis. So reclamation is therefore inevitable to for the geotechnical stabilization of the waste dump, accelerating the recovery of the post-mining ecosystem, generation of ecosystem goods and services, as well as sustainable land uses, and partially combating the global warming by enhancing the carbon sequestration process. So the planning of sustainable restoration program is closely related to the proper understanding of suitable vegetation species that can adapt that environmental stress and unfavorable mine spoil characteristics like high coarse fraction, high bulk density, acidic pH, low water retention capacity, low organic matter and nutrient content, etc. So in recent years, maximum from the restoration point of view, maximum global attention is augmenting in the sphere of selection of suitable plant species and its interaction with soil biota and understory vegetation, soil development, that is the pedogenesis process, microbial density and topographic heterogeneity. So therefore, suitable plant species selection is a critical factor towards successful reclamation of post-mining degraded landscape. But one more problem is this, that the impact of the vegetation approach on mine spoil reclamation cannot be evaluated by individual soil parameters, as most of them are interrelated and difficult to interpret. So the research is mainly based upon this hypothesis that the ameliorative properties of different plant species can influence the reclamation success towards forming a self-sustainable ecosystem. And to address this hypothesis, the aims of my lab scale experiments are like identification of specific soil quality parameters that influence the reclaimed soil health, development of CSQI, that is a comprehensive soil quality index for suitability ranking of plant species to the reclamation extent, and validation of this index with the vegetation characteristics like tree height, canopy cover, plant's relative density to confirm its functional utility. So my the field study was carried out at the 25 years old reclaimed OP dump in the Singrauli open cast project under Northern Coalfield Limited, Madhya Pradesh, India. And the study area was mainly reclaimed by the mixed plantation of leguminous and medicinal plant species like as a director indica, that is the neem tree, Dalbergia sisho, that is a sisham, Cassia shramia, and Terminalia arjuna. So the first and obviously an undisturbed forest land as a reference site also was taken into account, that approximately 20 kilometers from the active mining area to compare the recovery trajectory of the soil health with the post mining landscape. So during 2019, before COVID-19 pandemic, during 2019 January, the field study was conducted, and a phytosociological survey was conducted at this reclaimed coal mine site of 25 years old, and the plant diversity and heterogeneity was estimated by randomly placing quadrating method, and canopy cover was measured with the help of line intercept method. Tree height was by the help of distometer. Tree diameter, tree dBH measured at 1.37 trunk height, and soil sample was collected by randomly placing quadrating method and followed by coning quadrating method. So this is some field photographs during the vegetation survey and soil sample collections. After removing after collection of soil samples, the plant debris and coarse materials, that is greater than 2 millimeter coarse materials. After removing this debris and coarse material, the collected soil samples was divided in mainly two parts. One was stored under 4 degrees centigrade for biological parameter analysis, and the second was used for the physiochemical properties under this standard protocols. And followed by this analysis, CUSQI was developed through the PCA based weighted additive approach for suitability ranking of the plant species. Firstly, we analyze all the soil properties, followed by the selection of indicator parameters with the help of PCA. Then the scoring of that parameters with the help of nonlinear scoring function, and then nonlinear scoring function and the given weight was compiled to calculate the comprehensive soil quality index. So the rhizospheric soil under the leguminous plant species like Cassia and Dalbergia showed more improved restoration for more improved restoration capacity, like bulk density nearer to the reference forest soil than medicinal plant species like Azadirachta indica and Terminalia arjuna in 25 years old of reclaimed obidum. That could be due to the improvement of soil's texture, aggregate stability, soil organic matter input, soil and the interaction between the soil microfauna and rhizospheric activities that increasing with the revegetation age. In addition, the water holding capacity ranged between the 26% to 40% under different vegetation species, that could be attributable to the variation percentage of coarse fraction under the plant species. The moisture content of rhizospheric soil under leguminous and medicinal plant species in reclaimed mine soil varied significantly from 9% to 22%. That might be due to the dump height, type of plant species used for the reclamations, soil texture, soil organic carbon percentage, sampling time, thickness of the litter layer on the surface soil of reclaimed mine soil. The variation in pH also could be attributable to the litter decomposition, plant root exudates, carbonate-rich OB material, and also for the parent rock, parent material that underlying the study area, and amount of acid producing or acid neutralizing materials in the OB dump. The observed variation in electrical conductivity could be due to the variation in the root exudates of this tree species. The difference in the soil organic carbon, mainly due to the tree growth, higher litter fall, decomposition of litters into humus, microbial degradation, and organic matter input. Likewise, the variation in nitrogen, available nitrogen concentration in this technosol under different plantations may be due to the microbial activity, organic matter input, nitrogen fixation with the help of leguminous plant species, and organic matter mineralizations. The significant difference in phosphorus could be due to the soil substrate pH, organic matter, and weathering of the rocks. Obviously, similar variations could be seen in the rest of chemicals and biological parameters like potassium, CAC, DH, and microbial biomass carbon under different plant species. So in this to ranking the suitability ranking of the plant species, the total data set, like say 14 parameters, the parameters we are selected for the analysis are going for PC analysis, so principal component analysis, to select the minimum data set to define the critical soil characteristics that is actually responsible for the development of this index. Thus, from the PCA, we can select the parameters like for the MDS, like water holding capacity, available phosphorus, available nitrogen, and electrical conductivity, potassium, and dehydrogenous activity. With these parameters within each respective principal component, if there are more than one high loaded factor, then the weight were distributed between the correlated parameters, like correlation coefficient greater than 0.6. Neither the highly loaded or for non-correlated variables, the full weighted were assigned. So here is the correlation between the highly loaded variables from principal component analysis. And here is the equation formed from this PCA to calculate to develop the index. So to refine the functional utility of this CSQI, we form a regression analysis with the vegetation characteristics of that tree species that were mainly used, that were evenly distributed at that specific reclaimed mine depth of 25 years. That is the Azadirachta indica, Cassiasiamia, Dalbergia, and Terminalia arjuna. The canopy cover, the relation between canopy cover, DBH, height, and relative density with the CSQI was highly correlated, strongly correlated. That the functional utility of the CSQI is confirmed by these regression equations. And the CSQI observed for the leguminous plant species like Cassiasiamia and Dalbergia is higher, relatively higher than the medicinal plant species like Azadirachta indica, Terminalia arjuna, growing on reclaimed mine soil of 25 years old. That suggests the best suited vegetation species for mine soil reclamation in terms of recovery of soil health and ecological services. So based on the CSQI value, the tree species could be segregated into the three groups, like high CSQI, that is the best suited vegetation species, could be ranked 1, that is the Dalbergia sisho, the nitrogen fixer leguminous plant species. The moderate CSQI was with the rank 2, that is the Cassiasiamia, and low CSQI with the least suited vegetation species, that is the medicinal plant species like Azadirachta indica and Terminalia arjuna. So species with high CSQI value in present study could be recommended as most suitable for effective biological reclamations. And thank you for your attention.
Thank you for interesting presentation. We have time for one question.
Yes, sir.
Hello.
Yes, sir.
Ms. Bandyopadhyay, Cassiasiamia, an exotic species. On what basis, how you can recommend for best reclamation species?
So actually in this term, reclaimed OB term of 25 years, these 4 species are evenly distributed mainly. Cassiasiamia.
No, I'm not going on your soil features and all these things.
Yes, sir.
So that exotic species are not recommended for ecological restoration of degraded lands.
Yes, sir. That's why, sir, through the evaluation of the index, the study recommended the Dalbergia's issue as a highly recommended plant species with high index value.
But on the other hand, if the negative roles are given by exotic species, so can we consider on the basis of soil features?
Sir, exactly. These species are recommended with basis of soil characteristics under the rhizospheric soil taken from this site.
No. For ecological restoration is not a suitable species, Cassia semia. Okay.
Okay, sir. Okay, sir.
Okay, now it's time for break rooms.
Yes, so the breakout rooms will start just now. It will be for 15 minutes. And the rooms have names according to presenters, according to, and you have to choose your room. And there are 2 free rooms if you want to meet somebody and not talking about the presentation. So I start just now.
And we will meet at 10:45. Okay, yeah. 10 minutes later, basically, or 9 minutes.
Já jsem dal 14 minut vlastně, takže.
To je dobrý. Jak se sejde?
Jo, ono je to vyhodí potom samo.
Jo.
Hello, shall I start presenting? Wait.
Not yet.
Not yet.
As people are still in the breakout rooms, it will take
Ještě máme přestávku.
4 minutes.
Okay, thanks.
But we are happy to see you anyway.
Happy to see you too. Jak se máš? Dobře, to je dobře.
Paní Valmsle, chcete si to zatím vyzkoušet?
Můžu.
Když máme čas.
Jo.
Ty už máš 2 dítě, jsem slyšel, člověče.
Mám, no. Je to záhul.
To kotlík.
No.
F5 teď.
Jo. Už. Takhle dobrý?
Perfekt.
Děkuju.
Jo, super. Tak já to tam nechám. Nebo ještě to.
Možná, že bychom to mohli ještě schodit. To.
Jo, dobře.
Jo, aby.
Tam promítnem ten program, víš.
Jo, jasně. A co tvoje vnoučata, Honzo? Kolik? 2? Nepřibyli žádný.
Ne. To již jedině, když...
Teďka minuta, se to zavře. Tak, teď se to zavřelo, takže už by sem měli všechny vejít tady.
I see.
So shall I start?
So, okay.
Sorry.
No problem. So welcome back, and now it's my greatest pleasure to welcome Alena Wolmsley, and she will tell us something about utilizing of waste material to create soil for mine rehabilitation. So Alena, floor is yours. So
Thank you.
You can start.
Thank you, Peter. Thank you for the introduction. So I would like to introduce you to our research we're doing here in Australia at Monash University with my colleagues. and it's called From Spoil to Soil, or how to create soil or soil-like material from waste. At first, I'd like to talk, doesn't matter really. I'd like to talk a little bit about this area where we're doing our research. It's a brown coal basin that's got 3 large brown coal mines. It's called the Latrobe Valley, and it's about 2 hours, about 150 kilometers east of Melbourne. And yeah, this area has got a rich brown coal resource. And yeah, these brown coal mines are a bit different to the mines that we're used to in Europe. They've got a really thin layer of overburden, usually only 20 to 40 meters on top. And then the coal seam can be 100 to 200 meters deep. So you can imagine that it's a very, it's a very efficient type of mining. The mines are very prosperous, but the problem is that at the end there's a huge pit, and nobody knows what to do with it. So at the moment, one of the mines has already closed down and is being rehabilitated. It closed down in 2017 after this big fire broke out in 2014. The fire lasted for several months, and on the picture here at the bottom you can actually see this is the mine and this is the town nearby. Is quite a decent town, probably around 50,000 people, and yeah, everybody in this town has been affected by this fire quite a lot. They still suffer from respiratory diseases and stuff like that. So after this big brown coal mine fire, there was a big appraisal of mine rehabilitation effort, and the state government really put a lot of or started policing mines much more and ordered them to rehabilitate their land. Yeah, so this mine is now, they're now trying to decide what to do with the mine, and flooding is the obvious and easiest, most stable option. But there's an issue with water availability if they're going to divert the local river. And then it might affect the ecosystems downstream quite substantially. There's a lot of agriculture as well downstream, which can suffer from not having the water from this river. Yeah, so that's still not resolved until 4 years later. And then there's other 2 mines. So this mine on the photo is a Yellowhorn mine, and then there's Loy Yang mine, which I'm going to be talking about more because we closely cooperate with this mine. So here's AGL Loy Yang. It's the youngest of the 3 coal mines, and it's the biggest at the moment, and it's gonna be operating the longest from out of all of them. It provides almost half of the electricity for Victoria, or the coal-fired electricity, but there's not that many other sources, unfortunately, yet. And yeah, it's got 4 dredgers that operate 24 hours, and about 30 million tons of coal are produced each year. The pit is about 200 meters deep, and as I said, the overburden is only around 20 to 30 meters. And yeah, it's quite a large mine, and it's still getting bigger. But thanks to this mine fire and the following pressure that the government has put on mining companies, and the public as well has put on mining companies, all these mines really are really working on their mine rehabilitation planning and progressive rehabilitation, which until then wasn't so common or wasn't automatic like it is in Europe in most places. This mine will last probably until 2042, and the final void, the pit that's left, might be partly flooded, but it's going to be quite difficult because there's not really a water source, not a decent water source in the area. So there's a big emphasis on better stability, on stability of the slopes, so they can last without being flooded, so they are stable over longer periods of time. I'll show you some of these photos. So they're doing progressive rehabilitation, as I said, now on the northern slopes, northern and western slopes, part of the western slopes, and the mining is continuing towards south. The target ecosystem or target land use is pasture, which is slightly different from the two other mines. Around this mine, Loyang, there's mostly pastures, so that's why they're kind of returning to that land use. But also there's a big concern about the fire hazards, and grassland is considered less fire hazard than forest ecosystems, so that's also one reason. And also because there's not much overburden, not much material left, there's a big concern that trees could, so they only using 1 meter thick capping, and there's a concern that the trees could, their roots could grow into the coal and create cracks and then create instability. So that's also one reason why they're not really using trees. So at the moment they're using natural topsoil mostly, that they're stripping on the mine forefield, and the the overburden dump that's kind of behind the mine has already been rehabilitated. So at the moment they're just transporting material from one side of the mine to the other. And since 2017, 2018, they have been trialing artificial soil that we have been working on, which is very exciting. The main challenges in terms of soil and waste materials in rehabilitation is the utilization of the fly ash from the power plant, because there's a lot of it, and the fly ash from brown coal doesn't really have many good uses, as does fly ash from black coal, for example. So it's first leached in ash pond, and but close monitoring has to be ensured, so there's no leaching of salts and some other minerals that are not desirable in the environment. After that, it's stockpiled on the overburden dump, and that's where it stays, and there's really not a very good use for it, or yeah. no utilization. Then another waste material that's quite abundant, it's brown coal dust. It's just brown coal that's pulverized, and it can be used in the power plant to create energy. So it's just lying everywhere, and there's no good use for it. Then there's another challenge is shortage of topsoil, because the area of the mine of the resulting landscape is going to be much bigger than it was originally. So towards the end, they're very likely to run out of topsoil, and so that's why they're trying to find a solution to the soil scarcity. And another issue is erosion, the overburden, which is mainly consists of clay, but it's mostly kaolinite clays. It's highly erodible, and so is unfortunately the natural topsoil, which is very silty. And on the steep batters, it's quite often subject to erosion, especially in the initial weeks before grass is established properly. And another concern is weeds, because after the disturbance, the topsoil contains lots of seeds of weeds, and after the disturbance, the weeds tend to grow much better than the grass, unfortunately. So yeah, that's another issue. Now, the artificial soils, or the synthetic soils that we're designing and we're trying to optimize, they respond to two of these challenges. One is the shortage of natural topsoil, and the other one is utilization of the wastes. Also, we're hoping to reduce the carbon footprint of the mines, because our soils contain quite a lot of organic matter and have got high nutrient binding capacity, which should further increase the organic matter accumulation and lead to some carbon sequestration. And last but not least, this concept should form networks between industries, and hopefully even after our research is finished, these networks will still function, and these soils will be created during the mine rehabilitation. So what is actually artificial soil? It is it's constructed using industrial symbiosis. You probably heard that term. It's meant to be different industries kind of cooperate together, and one products from one industry are used by another one, and it forms like a cycle, which hopefully doesn't produce much waste. So in our soil, we combine the fly ash, as I said, from the power plant. This fly ash is quite alkaline. It's got quite substantial nutrient content and relatively high cation exchange capacity. Also, its texture is something in between fine sand and silt. And yeah, and so initially this material is mixed with overburden, which is mostly clay. It's quite acidic and doesn't contain many nutrients or organic matter. And by combination of ash and overburden, we create the base material that's supposed to have close to neutral pH, and shouldn't produce any leachate, any salts, or any harmful substances. And then to this we add some organic matter sources. We use a brown coal because it's got some stable forms of carbon, and it can also improve the soil structure. And then we use compost. Initially, we used mostly compost from the paper mill industry. And then we slowly transferred to green waste compost, mainly because of a silly reason that the paper mill industry waste had a bad smell, and the people, the technical staff didn't like that smell. So yeah, and the resulting mixture has got has got a loamy texture, so its textural consistency is optimal. It contains all the important plant nutrients, not always in the best ratios, but that's what we've been working on mostly. It has close to neutral pH in the ideal case, and it contains carbon in stable forms and has quite high cation exchange capacity. So our research has occurred in various steps. The initial step was analyzing the raw materials and trying to work out the best combinations. Then followed the laboratory lysimeter studies to check whether the soils are not leaching any harmful chemicals. And then we moved to greenhouse studies to test plant growth and to test how the soil perform in more natural conditions. And then we moved to field mesocosm studies. And then at the moment we're at the stage of field trials on the actual mine batters, as you can see here. We've had different issues during the process. I'm going to talk about them as we go along. I actually started my research with a field mesocosm study, which was focused on performance of these soils under natural climate. So with with natural rainfall that can be quite high, but can be very low as well during different parts of the year. And I have introduced earthworms as well to some of these mesocosms and tested how they survived and how they performed. And we tested plant growth. The results were not perfect, but we've proven that earthworms actually survive and you could almost say thrive in these soils quite well, although the pH was higher than we aimed for. The plant growth, however, was best in the base material, in the fly ash mixed with overburden. And later on we found that it was because the compost from the paper mill had really high pH as well. So it wasn't ideal for plants, but earthworms didn't mind it too much. Sorry. Sorry, I might just have to. I can't hear you very well. Can you repeat it again?
Time, please.
Oh, yeah.
Speed up a little bit.
Oh, speed up. Okay, sorry. So then we moved to field trial. And this field trial has similar problems as the mesocosm study. It had relatively high pH, and because of the drought, plants didn't grow very well. However, and we went back to greenhouse studies, and then at the moment we've got another field trial where we're testing new mixtures. Some of them still contain fly ash, but in lower amounts. And some of them, in some of them, we've introduced subsoil, or we've mixed it with topsoil as well. But again, at the start of this trial, the rain was very abundant, so that has affected the performance as well. Yeah, I'll just quickly run through this because, yeah, just the grass growth has been actually better in the artificial soils than in the topsoil, in the natural topsoil. On the other hand, weed biomass was generally very low. And soil chemical properties were quite favorable, apart from quite high pH, again, thanks to the fly ash present. But the other parameters were pretty good. Soil nutrients, again, in the artificial soils, nitrogen total nitrogen was high, available was similar as in topsoil, and phosphorus was much higher than in the natural topsoil as well, and potassium. So that was very positive. Now just briefly to the soil microbial populations. pH was the main driver, so the soils that contained fly ash had a relatively high pH, which most of the soil microbiota didn't like very much. However, there was quite a high diversity. Even the species, or even the richness was relatively high in some of the artificial soils, and the evenness was higher in the artificial soils than in the natural topsoil. Yeah, the fungal DNA wasn't, because we were sampling quite shortly after the experiment establishment, there was not much fungal DNA. And also probably due to the high pH, there weren't many fungi present. But yeah, this experiment has been started in 2020, so we
will,
yeah, next time I'll present some more results. And yeah, just some challenges we've bumped into when doing field trials. And the next time I'll tell you more results from monitoring of soil conditions at this field trial. And we're also planning to do some drought resilience simulation, because we've had a lot of rain, and we can get droughts for a few years and then lots of rain, and you never know what's going to happen. So thank you for your attention. And yeah, it was just a very brief introduction, so if you are more interested, we'll discuss it later.
Thank you for a very interesting presentation, and we will move questions to the breaking rooms. And now is my great pleasure to invite Professor Evgeny Abakumov from University of St. Petersburg. Please, you can start. Floor is yours.
Hello, everyone. Hello, Jan. Hello, Peter. Can you see my presentation?
Yes, we can see.
Yeah, we can see it here.
It is great pleasure for me to see you again. But we are online via Zoom. Unfortunately, we have no coffee break in real sense, and we have no Czech beer. only one consolation of this situation that we will contribute somehow to carbon offsets because we are staying at home. So my presentation is dealing with a microbial diversity of different aged contrast post-mining soils of Russian Northwest. I represent the group of co-authors, and I would say in advance that I cannot be in these rooms after the session because we have parallelly the session of Russian Soil Scientist Congress today. So unfortunately, I have another presentation, and I have to join another Zoom. So if someone have questions for me after this presentation, you can use this email. So I will start from our description of our current project, dealing with investigation of core microbiome of zonal mature soils of European Russia. You know that the zonality theory, zonality concept was established by pedology founder Vasily Vasilyevich Dokuchaev, and now we are aimed with the support of Russian Science Foundation to characterize the core microbiome of key zonal soils of European part of Russia. This caused only by logistic reasons because it is not easy to bring further soils from the Siberian plots to our research park and so on. And the important part of this research is investigation not only full profile zonal soils. These data are published already in Science of the Total Environment a few years before, but also to investigate the microbiome of anthropogenically transformed soils, which we have a lot of kinds in the territory of Russia. And the purpose of my group is to investigate the microbial diversity of abandoned mines and soils developed under the chronoseries of these abandoned mining landscapes. Here in these pictures, you can see examples of mining activity which are currently active in Russian Northwest, exploration of limestones, sandy gravels, granites and clays, which are quite unusual for quaternary surface of our landscapes, Cambrian clays and so on. And for this presentation and investigation, we have chosen two chronoseries. The first two chronoseries are presented by different ages from 3 to 10, 70, 60 years old of podzels formed on the abandoned Maluksa quarry of sandy gravel materials. And the second chronosequence is more long and more old and dated with a C14 carbon method. And both of them are presented in these pictures from 0 years, 20 years, 60 years, 70 years mature soil, which is presented by podzol, and another chronoseries, which is prolongation of the first one, located on the coastal bars of Ladoga Lake. You can see the youngest plot and the oldest plot, which is most remote from the coastal bars. So this is a quite good fast chronoseries of soil formations. Soil formation because soils are formed on the acid parent materials, sandy textured parent materials, which are easily to be altered and weathered under the influence of the coniferous plants. And the results of routine soil analysis of these chronosequences are presented here. And you can see that there is a differentiation of soil profile on alluvial type of podzol soils starting from the 30 years, acidification of the topsoils and removement of weathering products to the B horizon. So here, there is an initialization of podzolization. You can see small E horizon, embryonic E horizon, we used to call it. And on the 70 years, we have a prototype of zonal embryonic soil. and mature climax podzol in the age about 200 years. So we have extracted DNA. My colleagues, I'm not personally microbiologist and genetics of soil. I'm pedologist and soil chemist in my background. And we have traditional methods of PCR, Illumina sequences, and data processing. And what do we have in Chrono sequence? This is the youngest soil, this is the oldest one. We can see the increasing of bacteria, archaea, even fungi in the latest stages of soil formation. This in logic. These expectable and valuable results, I would say. Also, we have a slow increasing of fate indexes, number of phylotypes, hypothetic number of phylotypes in the community, and more or less stable Shannon index of microbial community. And as my colleagues suggest to represent data in more good way, they elaborated heat maps of soil microbial diversity on different depths and different horizons. And you can see that from the youngest soil to the oldest one, from the left part of each screen to the right side, for bacteria, chloroflex, and other phylums, there is a strong differentiation with the age, within the time, and within the depth. So the key conclusion of this part of my presentation is that the structure of core microbiome of soil genetic horizons is different within the depths and within the age. This is quite logic conclusion, but achieved and retrieved for the first time for these chronosequences in Russia. And there is some aggregation of beta biodiversity parameters within the soil age, and you can see easily that different horizons have different groups and aggregated finally in different spaces. And you can see that O horizons, B horizons, E horizons, and transitional emerging horizons of AY, which is called a dark gray humus horizon, are aggregated to different spaces while one analyzes the beta biodiversity. The older sequence of podzols, which are aged from the 70 to 1,500 years, also demonstrate the development of micro niches, micro-ecological, micro-environmental license with temporal scale. So we can conclude that the fast development of soil within the podzol soil chronosequence well corresponds with the diversification of soil microbial community. And this good support of Dokuchaev's theory that even soil horizon, not only morphological unit, but the genetical, biogeochemical, and environmental unit. And the second chronosequence which we have investigated is completely opposite in terms of chemical composition, because these soils are formed and derived from the limestones, crushing sieving remnants in the central part of the Leningrad region, where is Ordovician Plateau located, composed from the limestones. And you can see that very slow development of soil of Renzik type, with very slow vegetation development. And here we have not any intensive alteration of chemical soil properties. So Renzik leptosols, Renzik litosols stay more or less stable from 20 years to benchmark natural soil. And we cannot see essential changes in the diversity of microbiome. And only in mature soil under the forest with very well developed forest floor, we can see some changes. So going to conclusion, I would say that the investigation of these two contrast short and slow chronosequence on the limestones and fast and long chronosequence on the sands give us an opportunity to conclude that soil development rate is determined on initial stage by the characteristic of exposed heap parent materials, and mainly by physical properties and pH. So we are researcher, but not searcher. The searcher and scientist was Dokuchaev, and we are just a follower and those people who can contribute and confirm his hypothesis by new methods. So Dokuchaev formula and theory works, and horizon level well reflects the diversity of microorganisms, because every soil horizon is some kind of group of ecological niches and environmental licenses. So, and the last conclusion is a deeper profile means deeper microbial community penetration, which should be taken into account while one decision maker is elaborating the concept of reclamation in particular combination of parent materials and climate in various natural zones. So thank you very much for your attention. I believe I was in time.
Thank you for a very interesting presentation and for time. And we have time for one question, please. If somebody have a question.
And I want to invite the participants of this conference to contribute our young and developing journal, Biological Communications, the journal of St. Petersburg State University. Sorry for this advertisement and taking your time. It is indexed in Scopus, third quartile, not so high, but we are developing, and we have soil biology chapter in every issue. So if somebody will contribute us, and I know that Mr. Peter Hedenek is already have an experience. Thank you. So you are cordially invited.
Okay, so now it's time for another speaker. Fuji Kazumichi, please. You can start your presentation, Fujimi.
Yeah. Please wait. Can you hear me?
Yes, we can.
Yeah.
Perfect.
Thank you. I'm Kazumichi Fuji from Japan, FFPRI, Forest Research Institute. I would like to have a talk on the soil water retention in soil, including the coal mining sites. I'm from Japan, but my talk is mainly focused on the Indonesia. The Indonesia, East Kalimantan, the coal mining is destroys the forest, and this and the restoration is not so good due to the heavy clay. And you can see the very acidic water due to pyrite acid oxidation. The pH is around 2, and the childs are swimming around this. So I want to improve the soil and water both. So to improve the water, the soil should be the soil water retention should be improved. So today here I would like to analyze what is the key component for improving soil water retention. This is a very traditional soil water retention. For example, sandy soil has low soil water retention at across the pressure regime. On the other hand, the Japanese volcanic soil have the huge capacity of soil water retention. The coal mining soil of the heavy coal mining site with heavy clay texture have relatively low water holding capacity. So I want to know what regulating this factor and how to improve the water retention. The water retention, the to measure water retention, soil water retention, we need several steps. The water saturation and the sand column and the pressure plate like this, and the centrifugation and air drying and up drying. It's very time consuming, and I want to predict whether these specific devices. And today I would like to introduce the new approach to explain the soil water retention. The soil is 100%, and soil is composed of soil organic matter and the mineral fraction. And the mineral fraction is mineral fraction composed of sand, silt, clay, and the clay is further divided into crystalline clay and amorphous clay. So these components can be the players of water retention. So I did some kind of meta-analysis. I will move on to the results. And first, I fitted the all soil water retention curves into these very typical the equation. This is very difficult, but this is a very common equation, and this is the most general equation to fit the soil water retention, and we can get some parameter, the alpha and the n and the m, and describes the curve shape, and we can fit well. And after fitting, we can get the parameter n, but the parameter n can describe the uniqueness of sandy soil, but not in clay soil. So maybe it's good to describe the uniqueness of sandy soil, but it's not applicable to the all of the clay soil. Then I divide into this water retention by each at each the pressure head ranges. This decrease was explained by the soil components, and the multiple regression shows that the total water retention can be explained by increase with carbon, organic matter, but decrease with clay content. That means heavy clay limits water retention, and at the lower pressure, the clay carbon is very important in Japan. And the sample, due to the effects of Japanese sample, the volcanic ash derived clay also play important role in water retention. If we remove Japanese soil, still carbon is very important. And then clay also play important role, and the organic matter play important role at the higher water the pressure head. And then finally, the aerophane is very important. So to conclude, for initial saturated water content, the organic matter is very important, but the clay, too much clay reduce the water retention. And for Japanese sample, the aerophane is also important. So this is all the soil water retention curves, and the sandy soil is very, very bad, but the heavy clay also has the problem of low amounts of pore, and to retain good amounts of water. So the traditional description by Van-Genochten equation cannot predict well the soil water retention well, but our cumulative approach can be useful. So that means that if we have the basic soil data properties, we can predict the soil water retention without devices. It's promising. And even if we have the organic matter and clay, we can't doesn't always support soil water retention because there is no structure. So we need to identify what tree species increases soil building engineers such as earthworm or some others to increase the soil aggregates. So far, I have some data in the agent site. That is, we compared the various soil land use types, the soil carbon stock, and different land use type. And yeah, we can find that some trees, initial grassland, and then the plantation will provide the good amounts of organic matter in soil. But tillage always reduce the organic matter. So maybe the combination of these plantation and the introduction of some soil engineers are required to increase soil water retention and soil aggregate. Yeah, thank you. That's all, and thank you for your attention.
May I ask the question, or
yes, of course.
So basically, when you are speaking about overall retention, you are speaking about overall amount of water the soil can hold. But in clay soil, one can say that the curve is very flat. So large amount of water is actually under wilting point. So if we focus only on that part of water which is available for plants, that will be actually quite different, yeah? Am I right?
Yeah, exactly. What I measure is the soil water retention without plant. Such water budget in the ecosystem. If with plants, the more water can be retained in the ecosystem, for example.
Any other questions, please? If not, it's my great pleasure to invite another speaker, which is Professor Michal Bonkovsky from University of Cologne. And Michal, please, you can start.
Okay, so we heard this morning already on the about the usefulness of chronosequences as a general, big laboratories to study soil processes, and that's also our approach here. And my talk is based on this publication, and Jess Clayton was a lead author, was a PhD student doing most of this work, and so I want to acknowledge her here first. And so first of all, an introduction to our mining area. So you can see my pointer here. So that's Germany, and there are the sites, and here's the city of Cologne. And we have 3 mining sites, and I will talk about the Inden lignite mining area here, where we study the soil restoration after agricultural land restoration. And oops. So reclamation has a long history, and it covers nowadays about 300 square kilometers of mining area. So here's again the city of Cologne, and here's a kind of gradient of chronosequence, and we are now focusing on this area here. And this is how the mining site looks like. And generally, recultivation here, these mines, they move through the landscapes. Sorry, this is in German, but I will briefly explain. Yeah, here the mine activity starts. Here the overburden is taken away with these big diggers and then transported with conveyor belts to the other side of the mine, where agricultural land, in this case, is restored. And so the upper soil layer is here less material. This is in principle the dust from the ice age, and it creates the most fertile soils in our areas. And so the mining company is quite lucky that they have this less material to recreate soils. And so here on the right hand side you see a digger, how it takes away the soil, and here it's deposited. And so underneath it's tertiary sands, but on top of it there's a 2-meter layer of loose material that is shown here. So that is now the reclaimed agricultural fields, and so they have a 2 meters less layer, and this is how the agricultural fields look like still 4 years after mining. And so, and we study these chronosequence, and so it's because it's less material, it's very, very homogeneous. And since the mining company has not really changed its soil reclamation practice since over 50 years, this is providing a huge, laboratory to study soil development. So we have a high comparability of soils. And we study here, in this case, the soil development under intensive agriculture. And we also investigated the grass margins of these agricultural fields, because here we had a higher accumulation of organic matter. And we wanted to see the maximum that could be achieved in these soils. And so this is soil age. This is the soil organic carbon content. It's nitrogen content, extractable phosphorus. We will focus on this graph here. And this is, you see, with soil age, so within 10 years or so in the agricultural soil, so it's almost 1% soil organic carbon is reached, but it does not change much during the coming years. And in the margin soil, so on average 3% of soil organic carbon are reached within about 30 years, so it takes much longer. And so this gives about the potential those soils could form and could store carbon if we would manage it a bit in different ways. And so you see it can be even 4% in some places. And we also measured microbial biomass, carbon, nitrogen, and phosphorus, because our focus was on microbial stoichiometry. And you see it follows more or less the buildup of soil organic matter. The first thing we observed is that the young soils were highly variable in their soil conditions. So there's a high variability, for example, in soil CN content that is shown here on the left-hand side for field and grass margin. And so the variability decreased with soil age. So it was an indication that these young soils, of course, are kind of out of an equilibrium. And as the soils age, they got closer to a stable stoichiometric state. And so this brought us to the question, is there a threshold carbon or nitrogen value which determines the stability of soils, or where they reach some equilibrium? And now because our ideas have changed dramatically for the buildup of soil organic matter in recent years. So we have to acknowledge more the soil microorganisms. In former times, it was thought that the plant material mostly was the main source for the soil organic matter. And now the idea is that, well, the plant material has to be processed by the microorganisms, and the dead microorganisms and the microbial necromass, this is the main contributor to the stable soil carbon pool, because this microbial debris it attaching to the clay content of the soil, and this determines then, so the clay content determines the carbon saturation of soils. So that's shown here. So we have two fractions. So the stable carbon pool. So this is contained. This is formed by the microbial necromass, and it saturates as the mineral clay surfaces are fully saturated with the microbes. And this assumes that we have a very rapid increase in this soil organic matter formation, and then as the saturation is reached, then this will level off. And so the main message from these ideas about soil organic matter formation is we have to manage soil microbial biomass in order to increase and to understand better the soil organic matter formation. And so this can be done by microbial stoichiometry, because this determines the efficiency by which microbes use the nutrients to incorporate them into biomass and to produce then necromass. And this is, well, I would say the main figure of our publication. I have to guide you through this. So on the x-axis, it's the soil organic matter content in a log scale. And on the y-axis, it's microbial biomass. And in blue, it's microbial biomass carbon. In green, it's microbial biomass nitrogen. And in this yellowish, it's microbial biomass phosphorus. So these are the main nutrients in microbial biomass, so in bacteria and fungi. And so you see here different slopes. And so the steepness of the slopes, this determines the limitation of the microbes for a specific nutrient. So the steeper the slope, the more of these nutrients are acquired, and so the stronger the nutrient limitation is. And so we see shifts in this nutrient limitation of the microbes. First of all, we have a strong slope in microbial biomass carbon. So that's a slope here, 1.2. And it levels off at some point here with higher organic carbon content. And here where it levels off, we have an increasing slope of nitrogen. So it means, so the microbes shift from primarily carbon limitation to a carbon and nitrogen limitation. And interesting was that the threshold was about 1% soil organic carbon here. And we also have some transition area here. So we have some area with high variability where apparently the system is switching from one stable state to another stable state. And this is also seen when it's even better seen when we make the same graph here with microbial biomass nitrogen on the y-axis relative to total soil nitrogen. Yeah, so you see there's a really strong transition in the amount of nitrogen that's taken up in microbial biomass. And found that this has been something to do with the efficiency to incorporate carbon. And so we measured the maintenance respiration. It's also called the respiratory quotient. So that's the microbial respiration divided by microbial biomass. So you can think of it as the amount of carbon respired per unit microbial biomass in order to maintain the microbial biomass in absence of growth. Yeah, before they grow, yeah, just for the maintenance of the microbial biomass, they consume this amount of carbon. And here again, the soil organic matter amount in the soil, and this is the maintenance respiration. And you see that's dramatically high in soils with low soil organic matter carbon, and then it's decreasing, and there's also a break point where it levels off and is approaching some kind of constant rate. And this again is this 1% carbon threshold. And we now have evidence that this is probably related to the enzyme production of the microbes, because the microbes, they, in order to obtain nutrients and so from the environment and carbon, they produce enzymes, and enzyme production is very costly. So if you think of a small bacterium or so, and if there is no soil organic matter available where they can mine nutrients from, then this is extremely costly. And so this is probably relating into this really high inefficiency of microbial biomass production. And so this has major consequences now for the management of soil carbon in agricultural soils. So because you have seen in the agricultural soils, it's hardly overcoming 1% carbon. And so that means the system is always switching back to a very inefficient state where we have no accumulation of soil organic matter. And now we are starting a big field experiment where we increase the carbon content with compost above this 1% threshold, and we will see how this, whether our hypothesis here is really true. Yeah, so what does it mean if we assume that soil organic carbon is formed from microbial necromass? Well, we have to improve the efficiency by which microbial biomass is formed. And now coming back to this carbon saturation curve here that I showed at the beginning. Yeah, so we have quite good knowledge on this carbon saturation here at the upper end. Yeah, so there's some upper level where soils cannot store more protected carbon. But at the lower end, yeah, according to this curve, it's always concluded that the speed will increase or will be very high at the beginning to the carbon accumulation and then level off. But there is, Jan Froes actually published a figure here, and we also have seen a similar figure from Martin Bartuszka this morning, that the carbon accumulation is also slow at the very beginning of the when we have low amounts of soil organic matter formation. And what I showed you could be one explanation for this. So in order to conclude, so the potential for soil organic matter formation is not reached in the agricultural soils, so but potentially it could be improved. The young soils had a high variability, and as the soils age, they approach some equilibrium. And we found some microbial stoichiometric threshold at 1% soil organic carbon. That was critical for the, well, one could also say carbon use efficiency in the buildup of necromass, and this, which is a product for the formation of soil organic matter. And I have to thank the funding agency. Thank you very much.
So thank you very much for a very nice presentation. Now we have time for some questions, if you have. If no questions,
maybe say thank you to answer some of my question. I have got my results.
And now it's my great pleasure to invite another speaker, Bartolomiej Vos, and he will talk something about reforestation of post-fire soils. So please, you can start your presentation.
Can you hear me?
Yes.
Good morning. Okay. My name is Bartłomiej Woś, and I am from Poland, and I work in the Department of Forestry of Agriculture University in Krakow. I am glad to present my work about effect of tree species on carbon, nitrogen, and phosphorus stock and stoichiometry under reclined mine and reforested post-fire soil regeneration scenarios. Following large-scale ecosystem transformation due to anthropogenic, for example, mining and natural factors, for example, fire, the process of succession begins. It aims to restore soil function, macroelement cycling, and relation between elements. The long-term process of succession are often stimulated by human intervention through reclamation and afforestation with different tree species. One of the most commonly tree species used in afforestation in Europe are pioneers, Scots pine and common birch, due to low habitat demands. Competition for light and nutrients, especially nitrogen, are determinants of the succession process. For this reason, nitrogen-fixing species are also planted to disturbed sites. Under the European condition, alders are commonly used for nitrogen fixing during afforestation. However, alder finds its ecological optimum in sites with high fertility and humidity. For this reason, introducing alder to poor and degraded sites might result in different feedback when compared to its introduction to optimal growth sites. The aim of my study is determine the influence of pioneering, Scots pine and common birch, and nitrogen fixing. It is black alder, three species on the regeneration of carbon, nitrogen, and phosphorus stock and changes in soil carbon to nitrogen to phosphorus stoichiometry for post-mining as post-fire soils. My study sites are the reclaimed Szczakowa sand pit and Rudziniec post-fire site located in southern Poland. The reclamation treatment in post-mining sites consists of forming and leveling the surface, lupin cultivation, mineral fertilization, and afforestation. The fire in Rudziniec forest district, commonly known as fire in Rudy Raciborskie, and it was one of the largest post-war II fire in Europe. Directly after fire, the remaining trees were cut and post-fire site was replanted. The research plots we located in pure stands of pine, birch, and alder growing on sandy soils. Additionally, we set up control plots on sites with the optimal grow condition for the studied tree species. Samples from uppermost organic and mineral horizons were collected for physical and chemical analysis. The stoichiometry ratios were expressed as a mass ratio. The carbon stock in the post-mining soils were lower, and in post-fire soils similar to undisturbed sites. The effect of three species on total carbon stock was only observed for post-mining sites. The soils under pine and alder were characterized by a higher total carbon stock than the soils under birch. In the soils under alder, this was due to a higher carbon stock in mineral horizons. In opposite, in soils under pine, there was a higher carbon stock in the litter horizons. Similar to post-mining on undisturbed sites, we observed also the impact of three species on carbon stock redistribution between organic and mineral soil horizons. However, this phenomenon was not found on post-fire soils. The nitrogen stock in uppermost soil horizons was higher under alder in compared to other three species in all sites. Similar to carbon stock, the species influence the nitrogen stock redistribution between organic and mineral horizon on post-mining and undisturbed site. The higher nitrogen stock in litter was found under pine and in mineral horizons under alder. The phosphorus stock in post-mining soils and in post-fire site was lower than in undisturbed soils. During pedogenesis, carbon content increases due to decomposition of organic matter, while nitrogen content increases due to atmospheric deposition and nitrogen fixation. And phosphorus content remains relatively constant. However, this depends on the phosphorus content in the parent rock. For these reasons, the differences found in undisturbed soils result from the difference in a parent rock. The carbon to nitrogen ratios of the mineral soil horizon were lowest at the post-mining sites. The carbon to nitrogen ratio giving less than 20 leads to nitrogen mineralization and more than 30 to nitrogen immobilization. The carbon to nitrogen ratio indicated that the nitrogen content in topsoil was at appropriate level in the regenerated ecosystems. The highest carbon to phosphorus ratios and nitrogen to phosphorus were found at the post-fire site. These ratios indicate a phosphorus deficiency in the post-fire soils for a given content of carbon and nitrogen. The carbon to nitrogen ratio in the litter increases in the following order at all tested sites: alder, birch, and pine. However, this trend only transferred directly into the mineral horizons at the undisturbed site, which may be due to the shorter impact of organic matter on soil properties on disturbed sites. At the disturbed sites, the stoichiometric ratios indicate that the rapid accumulation of carbon and nitrogen under alder might have affected phosphorus deficiency and immobilization. The higher nitrogen to phosphorus ratio in alder litter compared to other species might indicate that the decomposition of alder litter is limited by phosphorus. The soil carbon to phosphorus ratios of 770 under alder in post-fire soils may indicate that this species strongly influence phosphorus immobilization. Although similar trends were also observed at the post-mining site, the impact of alders was weaker. However, this phenomenon was not found at the undisturbed optimal sites for alder growth. The carbon to phosphorus and nitrogen to phosphorus under alder was even lower, but not statistically in compared to soils under pine and birch. The different linear correlation of the carbon to nitrogen to phosphorus ratios between litter and mineral horizons observed for disturbed and undisturbed sites indicate the short-term impact of litter fall and that disturbance can decouple the carbon, nitrogen, and phosphorus cycling in soil plant system, a characteristic of forest ecosystems, stabilized forest ecosystems. And conclusion, the carbon stock of the post-fire site was similar to that of the undisturbed site, while it was lower for the reclined mine site. Three species had the strongest impact on total carbon stock in young post-mining soils. Three species affected carbon stock redistribution between the litter and mineral horizons. Soil disturbances can decouple the carbon, nitrogen, and phosphorus relationship. Alder adversely affected the carbon to phosphorus and nitrogen to phosphorus ratios in regenerated and undisturbed soils. Thank you for your attention.
Thank you for nice presentation. Please, do you have any questions for Bartłomiej? If not, I think it's time for breaking rooms.
And lunch.
And for lunch as well.
Yes. Hi, everybody again. So I will open the breakout rooms again. They will be open, the rooms will be open 100 minutes, so you can use them during the lunch. You can make appointment in any of them. And I see that Yevgeny and Fuji are missing. So these breakout rooms are, let's say, free. So I open and nice lunch and see you in the afternoon.
See you at 2 p.m. Central European Time.
Hello, everybody. If there is somebody who wants to test his presentation, you can do it just now and we can verify that it will work.
Okay, so may I test mine?
Yes, please.
I can't see it from...
Dole zelený.
Jo, já vím, ale nevidím tam tu nabídku.
Otevřenou prezentaci.
Otevřenou prezentaci a pak klikneš na to dole zelený a tam uvidíš fajly, který můžeš otevřít.
Není vidět?
Mám na výběr jenom plochu, whiteboard a iPhone, který nemám. A předtím...
Máte puštěnou tu prezentaci?
Mám.
Máš ji puštěnou na ploše?
Mám ji puštěnou na ploše.
A to je to z toho, jo, z prohlížeče.
Jako co?
Ten Zoom?
Ten by měl víc aplikace.
Jo.
A já už jsem to tam jednou viděla, když jsem to zkoušela předtím na nečisto, ale teď to prostě zmizlo.
Zkus to teda ještě jednou, úplně znovu. Zkus jako zavřít a otevřít tu prezentaci, vrať se do ní, zkus ji zavřít a otevřít. A pak to zkus celý znovu, jo.
Pavel, you want to try now?
Yes, if I can.
Yes, so try it because Petra has some problems, so try it now, you.
Okay, so I will click the share screen now.
Yeah.
And then share your presentation.
Find the right window and then click share.
Perfect.
Yes, this is exactly what you need.
Actually, have you seen only presentation on or
yeah, yeah.
You see a full screen, perfect.
Okay, so I stop sharing now. Thank you.
Yes.
You might try to switch it, just one. Yeah, good.
Yeah, okay, but it should work.
It's good. It's good.
Tak já se zkusím odhlásit, zase přihlásit zpátky do toho.
Možná no, já nevím, co to je teda.
Jo, můžu?
No.
Teda.
Yes, perfect.
A teďka F5 ještě. OK, yeah, perfect.
Tak jo. Tak, asi musím nějak
F5 se to pustí od začátku.
Jo, vím. Jo, tady to je. Tak.
Jo, takhle.
Nadějnic, ne?
Super.
Jsem ready.
Okay, still somebody else wants to test his presentation. He can do it. She can do it just now. No more. Okay.
Can I start?
It's time. Not yet. Well,
okay.
2 minutes.
Okay, perfect. I just wonder, would be possible add some timer?
Oh.
Just to measure countdown of 15 minutes limit.
Well, I'm not sure. I don't know how to do it.
Okay, no problem.
I was thinking about that, but I don't know how to do that. Well, maybe I think we can start.
Okay, so good afternoon for everyone. Hope you had good lunch or dinner, and now it's time to start afternoon session of our webinar. So it's my great pleasure to introduce Pavel Horodecký. So you can start, please.
Thank you very much. Can you see the presentation?
We can see it's perfect.
Okay, so let's start. Good morning, good afternoon, and good evening to everyone, wherever you are. And my name is Pavel Horodecký, and I work for Institute of Dendrology of Polish Academy of Sciences. And today I would like to present our result concerning humus layer development under various tree monocultures on a post-mining spoil heap. And at the beginning, I owe you some explanation because this title is a bit misleading, as I decided also to compare obtained results with those obtained in mixed stands. This research was financially supported by the General Directorate of State Forest of Poland. And we have conducted our research in central Poland, in Bełchatów Forest District, on external spoil heap of Bełchatów lignite mine that was afforested after the technical reclamation process terminated. And the area of this anthropogenic mount is almost 1,500 hectares. It was created till 1993 using material excavated from the cast before getting the lignite. And before being afforested, the surface, especially of the plateau of the mount, was covered by herbaceous, mostly end-fixing plants to create at least some of topsoil. And we decided to conduct our research on post-industrial areas as it is a perfect system to observe the initial soil formation. And as I believe, I don't have to convince all of you that the pedogenesis is one of the most important geological and ecological processes that determines the followed ecological processes. And its potentially high rate can shorten the time for crossing the threshold of self-sustenance of habitats, and especially their productivity, what is important for ecological but also economic reasons. And the assessment of particular tree species influence on upper soil layer development in such harsh habitats still need to be undertaken. Indeed, there are several studies that address the need of recognizing the post-industrial habitat potential. However, most of them are rather based on indirect methods. And this acquired knowledge can be used in new objects because post-industrial areas, despite their rather spotted occurrence, constitute in total quite significant area. For example, in Poland, it is more than 63,000 hectares of degraded areas, and due to the legislation, all of them must be reclaimed. And in the past, about 60% of reclaimed areas were afforested. It means that in Poland, about 38,000 hectares of new forests will appear in the future on soilless areas. And it is a perfect testing ground, perfect object to use our results. And finally, studying the soil development process on post-mining afforested areas enables the assessment of reclamation efforts, especially this biological. It gives us the applicational knowledge, indicating the tree species with the highest reclamation potential. And the aim of our study was simply to characterize the humus horizon of soils developed as a result of forest reclamation, to compare them with soil under the canopies of adjacent forest sites, and to assess simply the role of tree species type in pedogenesis. We hypothesized that the soil's humus horizon will be better developed in forest sites than on spoil heap. And the second hypothesis stated that the soil LA horizon will be better developed under end fixers and also mixed stands than other decidues, and also under other decidues than coniferous stands within both tested habitat types. And to obtain our aims and to verify our hypothesis, we have chosen 45 sample plots in total, and 23 of them were localized on post-industrial area and 22 on adjacent forest sites. And all the sample plots were chosen and paired, let's say, by criteria of herbal plant cover and also by soil homogeneity in terms of grainings, and with the same tree species that were in the same age. And between 2011 and 2013, we dug a set of 45 soil pits, and we determined a row of physiochemical properties. The soil pits were excavated under the canopies of 5 different tree species. It was black alder, common birch, Scots pine, English oak, and black locust. And 2 more pits we have dug under mixed canopy. And the stand age ranged from 17 to 25 years old in a post-mining site, and between 12 to 36 years old on forest sites, respectively. And the oldest stands were covered by black locust and the youngest by English oak. All 3 stands were divided into 4 groups according to their ecological properties. And we divided coniferous group, nitrogen fixing tree species, other deciduous tree species, and mixed stands. And fixing tree species, of course, black locust and black alder, other deciduous species, common birch and English oak. And samples were collected from the middle of topsoil horizon using standard cylinder samplers, 100 cubic centimeters in volume. And what have we measured was the organic carbon content, the total nitrogen content, of course, carbon to nitrogen rate, humus content. The P here is not potassium, it's a porosity, and this is the sum of the volume of soil spaces expressed as a percentage of the total soil volume. We measured also the bulk density and skeletal fraction content, and this is the particles bigger than 2 millimeters in diameter. And we also measure the clay, dust, and sand contents. Hygroscopic water capacity, it is the maximum amount of water that the soil can absorb from the air that is saturated with water vapor of 94%. We measured hygroscopic moisture. This is in turn the water that is unavailable to plants. And simply the depth of the A horizon, and finally the soil reaction. And what have we stated? In general, the soil A horizon in forest stands were richer in terms of organic carbon and total nitrogen content than on spoil heap. Nevertheless, the differences stated were statistically significant only for Scots pine in terms of carbon and for N-fixers when we consider the nitrogen content. In both habitats, there were also statistically significant differences in C and M concentration between species groups. And the A horizon, BNF N-fixers contain the highest percentage of carbon and nitrogen, while the coniferous the lowest. Humus concentration, that is highly positively correlated with both mentioned elements, displays very similar trends to them. And carbon and nitrogen in turn are highly correlated with each other, which is displayed by their rates that is very similar in humus horizon of all species group in both habitats. And C to N ratio ranged between 12 to almost 15 in coniferous stands. The soil porosity and its bulk density are obviously negatively strongly correlated with each other. And beneath the canopy of N fixers and other deciduous, we didn't state any statistical differences between habitat types. Oppositely, in coniferous stands, where the bulk density was significantly higher and porosity significantly lower on spoil heap than in forest stands. Both traits were very similar between species groups in forest size, while they differed statistically significantly on spoil heap. The hygroscopic moisture did not differ between habitat types among all species group, nor between species group within same habitat type. However, we could see that N-fixers favors its relatively high values. The maximum hygroscopic water capacity displays similar trends to hygroscopic moisture. However, some of the differences stated were statistically significant, like under the coniferous between habitat types or between species groups in forest sites. The pH differed considerably between species groups in forest size, but did not on spoil heap. And under the canopy of coniferous and other deciduous, the humus horizon reaction was significantly lower in forest sites than on spoil heap. However, this couldn't be surprising. And the depth of humus horizon was the highest beneath N-fixers, both on spoil heap and in forest sites. But nevertheless, all the differences stated were insignificant. The humus horizon granity was very similar in all group species in both habitat types. Nevertheless, we found some differences that were statistically significant beneath the coniferous stands in terms of skeletal fraction and beneath the coniferous and other deciduous in terms of clay contents. There was no such differences in relation to dust and sand fraction contents. They were pretty similar in all species groups. And the group of principal components with the eigenvalue higher than 1 accounted for almost 82% of the data variability. The first component explains 45% of variability, while the second almost 18%, and the two remaining 11 and 7% of variation respectively. And the main axis is connected with soil fertility, although most of N-fixer stands go according to this feature. They are not grouped at all. We could still can see some N-fixers stands on the left part of this axis. The second factor is connected with clay and skeletal fraction content and with the pH. And also here, N-fixers seems to follow this factor. However, also here, they did not group at all, as some of the stands of N-fixers stays in the left part of the axis. The third component describes more than 11% of the variance, and it had the highest loading on two soil parameters, negative on porosity and positive on bulk density. We could see that most of the coniferous stands, especially on spoil heap, grouped on the upper part of this axis. And the fourth component that crossed the border of significance is the C/N ratio. However, we could not say that some of the grouped species followed this factor in principal component analysis. The last slide is with conclusion, and we conclude based on our results that it's good to afforest post-mining sites with N-fixers, especially native, not with the alien Robinia pseudoacacia, and the most of all with mixed stands. We have to bear in mind that Alnus glutinosa, black alder, won't grow successfully everywhere. That's why we should propagate mixed stands instead of monocultures, as mixed stands can also accelerate the upper soil layer development. And our previous studies concerned litter decomposition gave similar conclusions. What is also important, mixed stands can also disperse the cultivation risks associated with the sensitivity of some species to harsh habitat conditions. Thank you for your attention.
Thank you for your nice presentation. Unfortunately, we don't have time for questions, so I will move questions for breakout rooms. And now it's time to introduce another speaker with Kristina Weissova, and she will talk us something about ecology and protection of post-mining industrial habitats from point of view of invertebrates. So Kristina, it is yours.
Thank you. It's okay?
It's okay, we can see.
Good day. My name is Kristina Weissova, and I am a student of doctoral program Applied and Landscape Ecology at the Czech University of Life Science. My project will focus on coal region and their transformation, which will be discussed in this presentation, and I worked on it with my supervisor Markéta Hendrychová. Due to the European Union's energy and climate concept, coal region must be prepared, reduce and close down mining activities. This will create large post-mining areas for restoration. This is a unique opportunity of preserve unique habitats and rare species of invertebrates. The aim of this project is to compare the connection of stinging bees and mold to the post-harvest landscape with the ongoing primary and secondary succession with reclaimed areas and to find a suitable combination to these methods. Similar to the methods, the collection on stinging bee insect and moths take place on 10 brown coal dumps in the Sokolovská and Severočeská basins. There are 3 study areas on each dump, reclaimed primary succession and secondary succession. Stinging bees are collected using colorful moeric traps, which simulate the color of flower. Preservative is poured into each trap, saline solution with a drop of spring. The spring here serves as a detergent, which reduces surface tension and accelerate the drawing of insects. The traps are left in place for 4 days, after which the sample are collected, stored in alcohol, and marked with the site code and the date of collection. Modes are collected using automatic light traps, which emit bluish light and attract animal with positive phototaxi. As the individual approach the diode, it hides the plexiglass and fall through a drain funnel into a collection vessel, where it gradually releases chloroform and puts the modes present to sleep. The traps are left in place overnight, then the sample are collected and frozen until determination. The results of the collection is so far suggest that ours with ongoing succession host more species and a large population of both representatives. The most obvious difference is in a rare and very rare species of stinging bees insect. When 20 rare species were discovered on succession areas and 12 rare species on reclaimed areas. Although the number of species and the number of individuals in reclaimed areas is lower, it is not negligible, and therefore it is very important to find the most suitable combination of these restoration methods to support biodiversity. This research will continue until August 2021. And in conclusion, I would like to thank for you the support of the internal grant agency at the Czech University of Life Science, without which this project couldn't have arisen. Thank you for attention, and please eventually question in Czech. Thank you.
Okay, thanks for interesting poster. And so any questions or not? If no, I would like invite another speaker, Karel Rehaut, please. Can you start? Oh. Is here, Karel? Probably not.
Yes, hi, I'm here.
Okay, hi.
Can you try to share your screen?
Okay.
Yes.
Green bottom. Yeah, perfect.
Yes, it's working now.
And F5.
Excellent.
All right.
Yeah, we see you great.
Yes, everyone can see the presentation correctly. Yes. Hi, everyone. My name is Karel. I work for the Canadian Forest Service as a forest biologist. Today, I will present you the results of a project I was working on while doing my master's degree. It is entitled Genetic Variability and Selection Pressure Influence the Phenotypic Responses to Stressful Environments, as well as bacterial and fungal assemblages in the rhizosphere of balsam poplar. Before I start, I would like to start by giving you some basic terminology to help you understand what I will present. So first, some basic mining terminology. For this project, we worked with two kinds of mine waste. The waste rock, which is the rock around the desired ore. Okay, yeah, like you can see here. And tailings, which is a waste product resulting after the processing of the ore in a mill. I will come back to those two mine wastes in more details later. Next, an important part of this project is the root microbiome. There are three main components of the root microbiome: the rhizosphere, which is the soil still attached to the roots after shaking; the rhizoplane, which are microorganisms directly in contact with the root; the endosphere, which is the microorganisms inside the root. And for this project in particular, we will only talk about the rhizosphere, which is the only component we sampled for this experiment. And finally, metagenomics. It is a study of the genomics, the DNA in the environmental DNA in a sample. So here, for example, it's going to be the DNA found in the rhizosphere. So basically, we have some environmental sample. We extract the DNA of that sample. Then we amplify a specific gene, which for bacteria is the 16S region, and for fungi is the ITS region. And then we sequence those DNA found in the samples. And we can obtain from those results, after analyzing, we obtain kind of like a barcode that will tell us which microorganisms can be found in our environmental sample. Now I will present the study sites. First, there is the Westwood site, which is a mine gold mine. It has a very large problem of acid mine drainage due to the presence of sulfur and iron. The waste rock here is coarse rock, so it's like you can see here, large debris. It is kind of orange due to the presence of iron. This mine site is still in operation, but the waste facility that I sampled has been abandoned for around 40 years. So you can see that only after 40 years, we can only see some tiny poplars trying their best to grow on that substrate. The other mine site is La Corne, which is a bismuth and molybdenum mine. It has been abandoned since 1972. It contains finely ground tailings. So like I said previously, the tailings is the result of, it is a waste that comes from the mill. So this is why it is so finely ground. And as opposed to the previous mine site, this substrate has a neutral pH, so it does not have this problem of acid mine drainage. Now I will present some results I obtained in a previous study, just to highlight why we obtained the further results in the other study. So we found that the presence of balsam poplars growing in the mine substrate on the site improved soil physicochemical properties. So it increases pH for the waste rock. It increased the presence of macronutrients, and it decreased the presence of sulfur and iron in the waste rock. We also saw a decrease in abundance of microorganisms linked to acid mine drainage, and the increase of abundance of microorganisms linked to nutrient cycling. And finally, an increase in bacterial alpha diversity. which is which represents a more healthy environment. And another finding that we had from this project is that we found that plant genotype can have an effect on the physical chemical properties of the soil. So here, what I mean by plant genotype is that we only have balsam poplars. So it's all the same species, but we sampled some different trees, and so we sequenced their genome, and we found that they are not clones. They have a different genotype, and even if they're all the same species, we still can find some differences in the soil around their roots. So, for example, in waste rock, we saw that there is less sulfur in their environment for some genotypes. So some genotypes can be better adapted than others for the restoration of mine sites. And we also saw that there are some genotypes that can better increase micronutrients. We also saw, like I said, an increase in alpha diversity in the mine waste, but we saw that in waste rock, some genotypes can have a bigger increase in alpha diversity than others. So like I said previously, this is an indicator of a better soil health. Finally, we saw that there is a difference in the rhizosphere microbial community structure. So those results are beta diversity, which means that each point represents a single tree. And the difference you see here is the further apart the points, the more different the more different their microorganisms attached to the roots. So what we see here is that there is a much bigger impact of soil type, but we still have a significant effect of the genotype on the structure of the microorganisms in the soil. And now for the current experiment I want to talk to you about. We saw that in the previous study, we saw that there was an effect of the genotype, but we only use genotypes that came from the mine waste of the study. So we wanted to know if we sampled genotypes that came from all across Canada, so with a much bigger genetic variability between them, we were wondering if we could see more difference between the genotypes. So here you can see on this map that there are three main genetic clusters in Canada. We have the Western genotypes, the Central genotypes, and the Eastern genotypes. And this genetic variability is a result of geography, which is abbreviated IBD for isolation by distance, and post-glacial recolonization routes, which is abbreviated IBC for isolation by colonization. So the genotypes that we had for our first experiment, we used them in the second experiment as well. And we defined them as two new origins contained within the central origin. So they are the Lac of Mine origin and the Westwood origin. And those genotypes are the result of selection pressure, which is IBD, which is short for isolation by environment. So the hypothesis here is that we believe if IBE is equal to IBD or IBC, so if the genetic variability induced by selection pressure is similar to the genetic variability in neutral population structure, and if genetic variability results in phenotypic variability, Then we should observe differences between trees of the three Canadian origins, Western, Central, and Eastern, as well as between trees within the Central origin, wild type, Westwood, and Lacombe. Regarding the rhizosphere microbiome, physical chemical properties of bulk soil, and plant growth. Therefore, we hypothesize that genotypes selected by the mine site environmental conditions are better suited for revegetation purposes than genotypes originating from natural settings. So what we did in that experiment is that we only used the waste rock. So we have 100% waste rock, 60% waste rock amended with 40% control substrate. And then we have a control substrate containing fertilizer. And what we obtain is, well, here you can see for the tree growth, after 2 years of growth in that substrate, all of the trees were very highly affected by the waste rock. As you can see here for non-amended waste rock and amended waste rock, compared to the trees growing in the control substrate. But what's very interesting is that in the non-amended waste rock, we saw a significant difference between the two origins coming from the mine waste compared to trees coming from natural settings. So we saw that genotypes selected by mine site environmental conditions seem to be less impacted by mine waste than genotypes originating from natural settings. And for the bacterial and fungal community structure, the results are much less obvious than they were in the previous experiment. So what we can see here is that bacterial community structure was only slightly impacted by tree origins. The only differences observed were in the control substrate and between genotypes from all across Canada, but we only have differences between the Western and Eastern genotypes. So yeah, the results are not that much obvious, like the points don't seem to cluster that much, but we can still say that the community structure in the Western genotypes is different than the ones we found in Eastern genotypes. And then for fungal community structure, we had significant effect in all substrate types. Well, except for the control, because the p-value is just above 0.5. But we saw that there is a significant effect of the origin in non-amended waste rock for trees within the central origin, and for trees from the central compared to Eastern. And finally, what's most interesting here is that we have the central genotypes, which are or wild type. They had a different community structure in their rhizosphere compared to the tree coming from both mine sites. So we can see here that the central are different from Lacombe and Westwood are different from Central, which means that the trees from the mine settings have do not select the same microorganisms, but the wild type select different microorganisms. Finally, for a conclusion, what we interpret from those results is that genotypes selected by mine site environmental conditions seem to be less impacted by mine wastes than genotypes originating from natural settings. The genetic variability influence bacterial and fungal assemblages in the rhizosphere of balsam poplar. And in conclusion, our results suggest that genotypes selected by the mine site environmental conditions could be better suited for revegetation purposes than genotypes originating from natural settings. In perspective, we believe that it would be best to have a longer term experiment in the future, because this experiment was only conducted during 2 years. Additionally, it was a greenhouse experiment, so a field experiment would probably give better results. And we would also like to compare our results to trees coming from nurseries. So thank you very much for listening to my presentation, and I hope it was clear.
Thank you for a very nice presentation. And now we have time for questions. Please, Jan Froos.
Maybe I have one question. You see many people try to supply seedlings from mining operation as much as possible. So there is, for example, a strategy to pre-stock them by phosphorus, but by extra phosphorus fertilization. However, the question is how long this hold when they came to harsh environment. So I actually like your approach to, choose genotype who are more adapted as a more promising. So can you kind of comment on that?
Yes, so what we understand is that previous studies show that genotypes that are not native of the environment could cause some problems. For example, inbreeding depression, which is some genes that are not adapted to the environment, should be meteorological components or the soil environment. So the native trees are much more suited for this kind of purpose. And as for the amendment, like you said, phosphorus, as the point of view of a microbiologist, I believe that microorganisms are much more suited than chemicals for the restoration of sites, mostly because, well, we know that phosphorus causes some problems like eutrophication of water. And if you use microorganisms instead, you can just put some phosphorus not in the ionic form like rocks, and microorganisms will just solubilize what the plant needs. So I believe that microorganisms are the best candidate for this purpose.
Thank you. Any other questions, please? If not, we can put another questions to the breakout room. And now it's my great pleasure to invite another speaker, Petra Benetková. So please, it's yours.
Thank you. Okay.
Yeah, we can see your presentation now. It's okay, everything.
It's on full screen. Okay.
Yes.
Can you see it now?
Yes, we can see perfectly.
So hello, my name is Petra. I'm from the Charles University in Prague. And it's very nice to meet you all, although I can't see most of you from face to face, but never mind. I would like to introduce you to one experiment that was conducted in or near in the spoil heaps of Sokolov in the Czech Republic. So most of the restoration projects are actually aimed on plant community restoration, which is quite understandable because we can see plants are visible. It's easier to control them, and also they are kind of source of primary production that we can exploit in case of meadows or forests. But from the scientific point of view, more recently, the bigger focus is, or the scientific. Well, we are more focused on what is going on below ground, and that by that I mean the microorganisms that, or the organisms that live in the soil. And it's mostly also because, as we know, the plants are affecting the soil fauna and vice versa. And also in the restoration experiments that are actually focused on soil fauna, the restraints are sometimes in the low number of taxa that are actually sorry, well, there is a low number of taxa, or the environmental conditions are quite limited in the experiments. So we tried to do, or was an attempt to do an experiment in the environmental, like the natural environmental conditions, and also we tried to sample as many soil taxa as we could. So to the actual experiment, the main ideas were to put an intact soil block of soil to maintain the biotic and the abiotic conditions as natural as possible. And sorry for that. And we also wanted to compare the situation right after the experiment and 20 years later. So there was a sampling in 1995 and in 2016. And the idea behind that was that soil blocks will provide refugium for the bigger arthropods and earthworms, so they will colonize the spoil heap faster and more easily than through the natural succession. How the experiment was made is like this, or the sampling was done in 6 plots. Well, the interesting fact is that in 1994 there was a landslide in the spoil heap, which exposed the bare substrate, which provided great conditions for this type of experiments because of the texture and the development state of the bare substrate. So into this substrate, there were 6 blocks of soil from nearby meadow transported and put right into the substrate. And the sampling was in the transported blocks, then in the plots that are marked or labeled as adjacent, which were 1 or 2 meters from the transported, and then the control plots, which were about 30 meters from the transported blocks. And the actual setup of the sampling points you can see in the left lower figure. And how the experiment actually looked like, you can see in these pictures. It's the whole process from the excavation of the block and transportation to manual reassembly of the blocks to make it like exactly as it was in the meadow, on the spot on the substrate. And the transported blocks in the spoil heaps are in the last picture, letter E. If we go to the results that you are expecting, we'll go from the lowest or the smallest organisms to the biggest one. So at first, in the left part of the screen or of the slide, you can see the results of the microbial biomass and respiration measurements. And as you can see, in both cases, the values were measured higher in the control plots than in the transported blocks, actually, which could be by grazing pressure in the transported blocks, and also supports the idea that in the control plot there is enough food supply for the colonizing organisms, so there is no scarce of food source for them in the new environment. If we move to the bigger organisms, let's talk about nematodes. In the upper figure, you can see a total abundance of nematodes in the initial period, which was the 1995, and the follow-up period, which are the green dots. And as you can see, the nematodes more or less follow the same pattern, and the values or the abundances are indicating that there is some sort of successful migration and colonization of the spoil heap, although the abundances did not reach transported values yet or by the time, but they are establishing quite successfully because the numbers also have risen by the 20 years. Below you can see the comparison between trophic groups in the sampling spots. What I think is interesting is the trophy group, the smallest one, the unicellular eukaryote feeders, which are considered or are connected with the later stages of succession, and apparently they did not reach the control plots yet. Either way, the others trophy groups were more or less not significantly different from each other. Let's go to mesofauna. Unfortunately, in the initial period, only oribatids were sampled. So you can see the result in the left part. There was a high number of oribatids in the transported plots, whereas very low numbers in the other two plots. But this pattern quite shifted in oribatids by the time. So in the follow-up period, on the right side of the figure, you can see that the numbers are increasing by with the distance from the transported blocks, which also can be supported by the enough food source by microfauna from the measurements we saw before. So apparently they are able to colonize and they are able to establish thriving community. And the other group that was studied from the mesofauna were springtails, unfortunately only in the follow-up period, but they were divided into 3 ecological groups. Within the epidaphic, within those that live on the surface, there were no significant differences, probably based on the fact that they are considered are strategics. They are quite fast to like colonize a new places, and they are not that dependent on the state of substrate of the soil as they live on the surface. So probably they just need some coverage and they're okay to move further. Then we have hemiadaphic and euadaphic springtails. There is different patterns in these 2 groups of springtails. The eudaphics that live in the soil were apparently able to colonize within the few meters from the transported blocks, as we can see that the number is highest in the adjacent plots. But apparently there was some barrier that was restraining them from moving farther from the plots. And the same pattern or the same case was in a group of hemiadaphic springtails, which the number were highest in, or the abundance was highest in the transported plot and was decreasing with the increasing distance from these block So let's move to the bigger animals. Now from the macrofauna, I chose two taxa or two organisms, and the first one are earthworms, and then the millipedes, Ulidae. As you can see in the initial period, both were present in the transported plots, the blocks, although millipedes in very small numbers, but they were missing anywhere else. By the time, if we move 20 years further, apparently the earthworms were able to somehow colonize the adjacent plots and even the spoil heaps, although the numbers were still considerably lower than in the transported blocks. And in the case of millipedes, they rose in the transported blocks. Or somehow they it was proven that for them the refuge is quite important, but they were also able to colonize the spoil heap, although again in small numbers. So if we are to sum this up, there was the good survival rate was proven in transported blocks, so it really it serves as a very good refugium for the organisms if we are waiting for the substrate, adjacent substrate to create better conditions. We were able to, well, there was we were able to create ideal biotic conditions versus unprepared abiotic conditions, which were in order to prove whether the migration barrier is bigger than the environmental barrier or the soil substrate barrier, and which proved that probably it plays more significant role than the migration barrier itself. That is something in the point below, that the soil development level is quite important when we want to get bigger animals that are considered environmental engineers to the site. We have to prepare conditions for them, and this importance is rising with the shape and with the size of the animal. And yeah, the transport of these intact turfs really did increase the potential to colonize the spoil heaps, but was not the condition that would grant the success in this colonization. And still, it's faster than an assisted succession here in the spoil heaps, which we can see from the another studies. And that will be all from me for now. So thank you for your attention. I guess we have some time for
thank you
questions.
for the interesting presentation. And now we have time for questions. Okay, so please ask. Okay, I have just one technical question. If you were extracting nematodes, did you find also some NH traits? Because I can expect you can find a lot of them in.
I would have to look into the original datasheet. Perhaps yes, but I really can't tell you right now.
You can move it to the breakout rooms. So now it's my pleasure to invite another speaker, who is Fabio Vicentini. So please, Fabio, you can present your data.
Oh, so okay. Thank you for letting me speak now. Thank you for inviting me. So that I guess I will share the video, share the screen, and I've never done it before. So let me.
If you
just,
have voice in video.
Do you hear me? I say, is it okay? You hear and you see what I'm seeing now?
Yes.
Yes.
I don't know it entirely. So, but I will continue then from here. Let me just. I'm also. Or well, I'm also really rusty in just using these basic tools.
So try maybe F5 if you click on F.
It could be the easiest one, actually. No, if it wasn't that mine doesn't do it. Yeah, there we are.
Okay.
Thank you very much. Okay, so let me. Okay. Okay, so post-mining sites offer a unique opportunity to investigate environmental processes due to a synchronic presence of different stages of soil succession in a restricted area. The procedure to preserve and implement ecosystemic value that the post-mining sites offer is a topic often open for further investigation and discussion. What happens that the leveling or the dosing of the heaps of burden left behind after a mineral extraction is a common practice that makes place at the start of post-mining sites restoration. As a result of this intervention, the microtopographic heterogeneity represented by the heaps forming waves on the land is typically suppressed, reducing the landscape to the leveled area, determining a reduction of the possible environment available for plants and fauna. Many studies report similar results that we are going to present in this in the subsequent minutes. And often presents a reduction of calamigrostis, invasive plants, and an increasing of the available niches for the species. The study was conducted in the most mining area in Czech Republic, considering the map. There were two chronosequences. It was investigated. In both chronosequences, no trees had been planted. The first chronosequence consisted of sites where no leveling or any other technical measures were taken after heaping, and sites were kept with their original waves with no changes afflicted on them. The second chronosequence was formed by sites which were leveled by earth-warming machinery, warming would be nice, earth-moving machinery, and in most sites, some shallow topsoil layer was spread. Both chronosequences were about 30 years old and consisted of from 8 to 11 sites. In addition, a birch site outside of the heaps was sampled as a control. And inside the sampling plot, where 10 per 10 meters was established, located at least 5 meters from the site margin. Plot age was determined based on historical data. And what was collected was both meso and macrofauna, and also some samples for chemical analysis. They composed of 5 individual subsamples of 100 grams of topsoil up to a depth of 5 centimeters below the layer, the little layer, in March of 2004. So now we go to the results. What we have found. Development. The first one is about the development of vegetation cover with the site age on the x-axis. It was different between grounded site and sites from the original wave surface, where the original wave surface was maintained. In a flat grounded areas, there was a significant higher herb cover. Moreover, herb cover in these sites was high already during the initial stage and slightly decreased with site age. On the contrary, no herb cover was present at the beginning in wavy sites, and it decreased throughout time. Consequently, the difference in herb cover between flat and wavy sites decreased in the older, later stages of succession. The woody cover was significantly higher in wavy areas than in leveled ones in all ages of the succession. In flat areas, the wood cover increased slowly in the last stages, whereas in wavy area, the increment started during the younger phases and continued in an unoscillatory pattern. And we go to soil chemistry, were similar in both types of overburden management. The pH in both graded and undegraded area decreases slowly with site age, being generally more basic in the younger sites and becoming more acid in the older ones. The available phosphorus was not affected by site age or leveling. Sodium concentration was significantly decreased by the site age, and there was no significant effect of overburden grading, although it remained in either concentration in wave areas than in flat ones throughout this time. And we go to the fauna. And none of the fauna groups that were studied were significantly affected by either age of surface leveling. In most groups, the site in wavy underground sites showed a tendency of increasing abundance over time, while the graded site maximum abundance could be found in sites of heavy or middle ages. Different parameters between flat and wave areas were more visible on the PCA diagram. Oh, that should be, I deleted it, a funny thing. So, sorry, I actually don't, can I find it easily? Well, there should be more, let me go out, how can I get out of that? New share.
Okay, skip it and go on, I think.
I actually have it as thing, so it should be, I go pick it up from the article. Yeah, so you see it here. So the PCA, where it should be more visible in the PCA, where age and type of substrate are considered together. We can see that there is a clear distinction between level and wavy size, although there is no clear effect of time. The earthworm and terrestrial isopod had a tendency to be more abundant in wavy side, while millipedes, fly larvae and centipedes were found in greater density on level side, on leveless side. Okay, so well, now just to the conclusion, the surface leveling supports a dominance of competitive species such as an expensive grass Calamagrostis. Calamagrostis becomes more dominant throughout the ages, reducing the potential for trees to root and establish, while the non-reclaimed wavy surface were capable to reach a richer and higher heterogeneous environment than might be hoped for in conservation plans. So proving that intervention can be good. The suppression of topographic heterogeneity by surface dosing affected negatively the studied fauna group. Such outcomes support the positive impact of the larger scale habitat heterogeneity on biodiversity in post-mining sites. And with this, I conclude. Yeah, that's something. I think I would just. So yeah, I'm not hearing any comments, so I'm not even sure if the last part got through, but. Yeah,
So
you are
that's okay.
sharing.
So
stop share. Yeah.
Okay, so do you have any questions?
Yeah, maybe.
Okay, so Mikhail, please, you can ask.
So this is then mostly showing the trade-off between a kind of arrestment of succession with the Calamagrostis grass and then the succession of forest sites. That's right, huh?
No.
Well, this part I will not collect and say it's not.
Can you hear me?
Okay. Yes, I'm sorry. Okay, so you were pointing out about the vegetation and another point.
Yeah, so it's a trade-off between the arrestment of vegetation, yeah, by your Calamagrostis grass and the forest sites. Did you also try, for example, some regressions then? But because in the non-forested sites, it was a very clear pattern that the succession set on quite late. And did you try to correlate this, for example, with the appearance of woody vegetation, so that's this should give probably.
If I remember correctly, we did something about it, but nothing was showed of significance of that was relevant to be included into here. So that I unfortunately cannot be more detailed about it because, well, I picked it up when it was already started. So many details about it, I cannot actually dwell into them.
Okay,
so,
thank you.
Yeah.
I might comment on that maybe. So basically in this particular study, we cannot really show that relationship statistically because we don't really have large number of sites. That was the major problem. But we did a previous study, which was actually inspiration for this, and that was experimental. So we have 3 sites which we leveled and 3 sites which we deep graded, and we follow them for 12 years. And we can say that on the graded sites, there was just a Calamagrostis, and in the non-graded sites, there was a tree establishment. And there is clear trade-off. And we believe that this is actually related to soil compaction and to elimination of the safe spaces which allow tree to establish. So it's a several factors. It's a Calamagrostis competition, higher compaction, and elimination of safe spaces, which actually make tree development, slower in the graded sites.
Okay, more multifactorial, more difficult. Yeah, thank you.
Okay, any other questions? So thank you again, Fabio, for a presentation. And now it's time to introduce another speaker, Yamile Dominguez Haidar, and she will tell us something about the role of ants as ecosystem engineers in post-mining areas. So please, you can start your presentation.
Thank you. Good afternoon. Moment, please. Okay, and good afternoon. I'm happy to stay here and know all works. I believe that in other conditions was impossible share this work all us. My name is Yamile Dominguez. I'm from Universidad del Atlántico, Colombia, and I'm here to share the work impact of Phaedolephallax Hymenoptera formicida as ecosystem engineers in rehabilitated coal mine areas. Okay, the efficiency of rehabilitation can be evaluated, measured chemical and physical soil parameters, and also the reconstitution of the soil macrofauna community. In the mine context, ants and particularly important regulators of soil processes are considered ecosystem engineers, and basically for different processes that they can lead as change soil physical properties, generates bioturbation, affects porosity and water flow, and modify the chemical composition of soil by incorporating nutrients and organic matter. In special, Phaedolephallax, it's a predatory and carrion ants with a neotropical distribution. The nets have galleries that lead to several chambers near the surface on the ground, with the refuse piles on the outside. We selected Phaedolephallax in rehabilitated areas of the Cerrejón coal mine because in previous study, this ants was the most abundant in this site, especially in sites with more time of rehabilitation and forests, and also was identified as important seed disperser. The objectives were test whether Phaedolephallax nets have an effect on soil chemical properties, characterize the debris of seeds, arthropods, and other organic items found in the refuse piles, and describes net architecture as a proxy of bioturbation effect in the soil. The study was carried out on Cerrejón coal mine, northern of Colombia. The natural ecosystems surrounding the mine is seasonal dry forest with bimodal precipitations April, June, and August, December, and high evapotranspiration. The rehabilitation protocols used in the mine consist of three principal stages: land geomorphological adequacy, topsoil stabilization with grasses, and revegetation with native species. We selected in this mine 2 areas with different rehabilitation lifetimes, 16 and 20 years old. 3 plots in each area were established to locate nets of phaedolephallates. For this, tuna baits were placed in the soil in each plot in a 3 time 20 meters grid. And for characterization of biomass and organic debris of refuse piles, 5 nets per plot were selected randomly. All debris found in the refuse piles was collected. In the laboratory, the organic debris was sorted into 3 groups: arthropod debris, seeds, and parts of other animals. Each sample was cleaned, dried, weighted using an analytical balance. This is an example of the refuse piles. Chemical properties and near-infrared spectra analysis were assessed only for 20 years old sites, given availability of nets at the moment of this survey. 7 nets were collected and 3 types of samples were collected. One is the interior of nets, the other is refuse piles, and control samples 1 meter of the nest entrance. To evaluate ability of NIRS and chemical variables measures was this. And to evaluate the ability of NIRS to discriminate net soils from surrounding soils, 10 grams samples were taken from the same points and sieved them through a 500 micrometer sieve. 1 gram of each sifted soil was scanned with a spectrometer in this spectral range. Measures of absorbance were taken at 8 nanometers intervals. For evaluating nest structure as a proxy of bioturbation, the soil removed for construction on the nest was estimated based on the net's volume. For this, 2 nests from 16 years old and 3 nets of the 20 years old sites were selected for description of the internal structure by 3-dimensional models obtained by impregnation with dental gypsum. The gypsum molds were excavated 2 days later by digging 1 meter deep from the net's entrance. The molds were measured to determine total volume and total height. For data analysis, items of organic debris found in the refuse piles in each area were compared with a non-parametric Kruskal-Wallis test and subsequently post-hoc done test with Bonferroni adjustment. Biomass, and to compare biomass with, between areas, a non-parametric Friedman test was performed. The item was used as blocks. A Kruskal-Wallis was test. A Kruskal-Wallis test were used to compare chemical variables among interior nests, the control soils, and the refuse piles. For NIRS, measures to absorbance were transformed to second derivative. Then we performed a principal component analysis, followed by a discriminant analysis for significant differences among these factors. All analyses were done in AD4 in package R. Okay, the results, the refuse piles of both sides were composed mainly of arthropod debris. This was greater than other items in the 20 years old site. But in the 16 years old site, the difference was barely with other arthropod debris. The biomass of the items in 20 years old site was greater compared to 16 years old site. Most debris observed corresponded to carcasses of Coleoptera, Hymenoptera, and Orthoptera. Seeds, principal of grasses and legumes, were the second most observed items in the refuse piles. The most frequently collected items composing the other animal debris were pupa, herbivory feces, and mollusk shells. Data obtained from external refuse piles suggests that Phaedolephallax colonies use foods from different trophic levels. The greater amount of arthropod and seeds found in the refuse piles in the 20 years old area compared to 16 years old area could be indicate greater supply of resources, which would be related with a better habitat condition, favoring the establishment of greatest number of nests. An example of seeds found. For chemical analysis, refuse piles, samples of Phaedolephallax were different from the net and the control soils. However, the interior nests not different from the control soils. The content of nitrogen, phosphorus, potassium, magnesium, and organic matter was greater in refuse piles compared to soil control. This was 14 to 70% here, greater. And this tendency, the difference between refuse piles with the nest and control soil, we can observe in this PCA with the results of NIRS. The separation of the refuse piles from the control nest and soil is observed here. In this case, NIRS, to difference this, can separate nets from control soils. If you can see, the very centers are separate. And the first components separate the sampling plots and show high variability in between plots. The second component distinguishes the soil's origins. The refuse piles formed a separate group. NIRS is a practical technique that allows distinguish refuse piles from the soils in the interior of the nets and soils surrounding the P-phallax. And not only for P-phallax, these techniques is usually important to distinguish these fingerprints that macrofauna do in the soil. The hauling of this debris and seeds could trigger the incorporation of nutrients into the soil, causing an enrichment on the adjacent edaphic horizon. In other words, high values of organic matter show that this part of the net is an important source of accumulation, translocation, and transformation of soil organic matter. Non-difference between net and control soils is possible due to mixed soils or by bioturbation. Final, for next architectures, the net molds presented an asymmetric architecture. The chambers have irregularly shaped edges. These chambers are interconnected with wide horizontal and vertical tunnels. Net height ranged from 5 to 11 centimeters. This height is lower is compared to other species of the same genus. This may happen because rehabilitated areas have a soil layer the 30 meters, more or less. Each would be a limitation to building deeper nets. Another possible explanation would be related with the size of the colony. Although this was evaluated in this work, based on the previous study in the same area, we found that a high rate of turnover in the nets. And we believe that the fact that the nets don't reach larger sizes, yes, caused by turnover, high turnover. The net has a total volume ranging from 30 to 106 cubic centimeters. If we connect these results with other results that assess density of nests in this size, we estimate that the moment of the work, this remotion. But if we consider that the high turnover in this site, we believe that this remotion can be doubled in different forms. Finally, as conclusion, we can conclude that the phaedolephallax plays an important role in the reestablishment of ecological processes such as soil organic matter accumulation, seed dispersion, and bioturbation. Further studies are required, in special with a more complex quantifications of the bioturbation impact. Thank you so much.
I have a question. May I answer? May I ask how many?
Sure. Okay, yes, you can.
Sorry. How many ant species do you have there? And so this is just one of them. And so I guess there are many more ant species on your forest floors?
Yes.
Or is this the most important one?
Okay, the previous study showed that the richness of ants increased with time of rehabilitation. This site have lower species related with a forest, but the composition is more related with the forest. In the forest, I remember more or less 50, more or less. And these sites have 30 species, more or less. But in special, this species, and Ectatoma ruidum, was the most abundant in the shared sites, forest and sites with more time of rehabilitation.
Thank you.
I have two questions. Did you try to compare this bioturbation effect also with natural habitat, like in this forest habitat? That's one question. And the second question is concerning the seeds. You said the seed transport is important. Did you try to test if the seeds in the refuse pile can germinate, if they can be source of another plants, basically?
Okay, thank you. For first question, no. This is the idea in our next work, compare bioturbation in forest. Indeed, the idea is estimate a rate in the time, because was only an approximate. But it's important, and we try that in the next work measure this. And since, yes, in a previous work, we determined that the seeds carried to nests and the refuse piles can germinate. And germinate is higher compared to control soil, because we consider that this species is important to in this process, the dispersion of these seeds. In special, we found that can disperse seeds from seeds of native species.
Thank you.
Okay, so any other questions? If not, I will invite the last but not least speaker, Amir Haas. Please, it's yours.
Thank you. Hi, you see me, hear me?
Yes, yes,
okay.
Perfectly.
Okay, I'll leave this and that. Okay. Right. So I'm sharing the screen. Can you see the presentation?
Yes.
All right, perfect. So first of all, thank you for having me. And it took me a while just to understand the nightmare of trying to, for you guys to coordinate this over so many time zones. So yeah, don't envy you. At any rate, I want to share with you some finding that we recently we're working on the mine site, and we're in West Virginia in the United States. And basically to give some insight on the role of redox promote dissolution processes that we think that a little bit are overlooked in this environment. So a little bit background, the way that we're practicing here in West Virginia in the Appalachian in general, it's what we call a mountaintop removal operation. There is a provision in the legislation that actually allow a mining operation to use the overburden or that crushed, blasted rock material as a topsoil replacement. And the question early on in the 2000s was, should we use the more near surface brown sandstone, which is more weathered, of a regolite type material, if you will, or use the more deeper gray sandstone that, and the distinction is between the colors with the more oxidized brown materials that is the near surface versus that gray. And you can see the plots here as they're being laid out. You can see here, let me just pull that. Okay. So we see in the foreground, we see the brown sandstone plot being kind of pushed around. And in the background, you can see the gray sandstone plot. The site was established in 2005. And you can see here from that satellite image, that's the layout of the site. In 2017, we came and we basically instrumented the site with some shallow observation wells. And here is what the pins represent, those different wells at those different plots. So we have the gray site, which was then split into compacted and non-compacted treatment. And we have the brown sandstone site, which again, compacted and non-compacted split. And the compaction was basically kind of the common practice at the time, where the non-compacted were practiced with as least doser passes as possible during the spreading of that overburden material. Let me share with you some of the results that we got. Going to be a little bit busy here. We're looking at different parameters that we were looking at. And these are reference values. So we see mean and range of what we'll expect in surface water in our region. And this is what we find in this study. And to make this busy table a little bit more palatable, we can look at the ratios, basically at the values that the average value that we got in this study compared to the mean of that reference benchmark. So a value of around 1 will mean that we are at par or not exceeding what the reference values are. And we can see that as far as pH, total dissolved solids, and alkali and alkaline earth metals, we're well within that range. We do see that, that in as much as we're looking here at manganese and iron, we're 2 to 3 order of magnitude higher as far as soil water chemistry is concerned compared to what surface water benchmark will be to our area. So that kind of already gave us an idea that these are redox sensitive element compared to some of those magnesium, calcium, and potassium, of course, which are not that redox sensitive. And then we look, so here we're looking at the total dissolved solids, or the EC, the electrical conductivity, as a function of pH. And we see here the different point measurement that we have from the brown side. Those are the brown symbols here. We can see the ones from the gray side. We also have this open green ones are a native forest nearby that we collected some sample from. And the blue ones are surface water from the brown sandstone site. And what was intriguing was this kind of a triangular shape with those higher level of total dissolved solids actually occurring in a circumneutral pH. Now, EC is a very, and I assume in your all areas as well, is an easily measured, very cost-effective parameter that can be retrieved in real time and really quickly, and it's adopted by the regulatory agencies, at least in the US, as a threshold for water quality. And I'm putting here some of those reference values of 5 and 300 microsiemens per centimeter. And you can see again that we're exceeding that water quality benchmark, and that exceedance occur in circumneutral pH. And this was a little bit surprising because we actually were expecting that high levels of dissolved material will occur and will be associated with some acidity and some acid promote dissolution processes, especially as we're, you know, we have the legacy of pyrite and sulfur minerals, and acid promote dissolution and is almost the process du jour in such systems. So that whole idea that we actually see those high levels in that circumneutral pH was a little bit of a intriguing findings. If we further look at the redox potential here, at the same, also against the pH, and we can see the brown sandstone points, and we can see the gray. And again, we can see that this one gravitates also. We're getting the lowest redox value as we're going towards circumneutral pH value. This is much more easily can be explained given that microbial activity and respiration and increased levels of CO2 that put kind of a downward pressure on pH, if you will. And then, of course, redox processes that actually consume protons have an upward pressure on pH. So in a well equilibrated system, like in wetland or swamp, for example, you do see that at equilibrium, we do find that those systems tend to have a neutral pH system. Here we just superimpose both of those data sets. So we're looking at the EC and that redox potential, just to, again, to kind of try to provide a visual presentation of that mirror image. We then just did a bit cleanup of the data. We just averaged them on a half a pH unit interval. And this is what we see from that study. And again, we're talking about 3 years of study where we're sampling on a weekly basis throughout the growing season. And this is the accumulated data set that we see. And again, we're looking at EC as it actually, excuse me, the redox potential as it decreases as pH reach circumneutral values. And then if we look at the EC, or the total dissolved solid, we actually see that peak at that same pH level, and it is associated with that low redox potential. And that again kind of gave us the whole conceptual framework that something is going on here. And in fact, those values that exceeds those regulatory threshold and kind of deem that water to be somewhat of a concern in those ecological systems, to actually suggests that there is something associated with redox rather than pH, because we are talking about some neutral pH value. And so it led to some hypotheses, and the hypothesis was that maybe some terminal electron acceptor and their abundancy may affect and govern the actual redox potential. And here we look at that redox ladder. So of course, oxygen is usually consumed at it's the most energy efficient terminal electron acceptor. But in a system that once this being diminished and being consumed, of course, the microbes of population that can consume the next more energetically efficient terminal electron acceptor will strive, and this will be nitrate on a kind of a big picture, and manganese oxide and iron oxide and sulfate. And of course, there's some overlap between those, but that's the idea. And we thought that it might be that if we have excess of one or the other, or maybe we're missing some of those, then we'll have this ladder kind of broken in a way that may help us explain why we see redox value to be affecting so much that stability and some of those parameters that are being that those sites are being judged and evaluated by. So we were looking at those three material that we have, the gray sandstone material, the brown sandstone, and that native soil. And we are basically introduced in a suspension incubation study in a glove box, either nitrate, sulfate, iron oxide, or just a control that didn't have any terminal electron acceptors. And we conducted an anaerobic incubation study. Basically, they were putting in a jar, a sealed jar, and we let those condition developed, and we looked at those redox potential over time. Here we're looking at the gray sandstone. So again, this is incubation time, and this is the redox. And of course, we see this decline as oxygen being consumed, and we see that redox decline, but we really don't see any treatment effect, meaning with none of those terminal electron acceptor actually result in any mean separation that suggests that they actually affecting that, that system. Here is the brown. Again, we see some initial separation, but again, not something to write home about. And in fact, in the native soil, we finally see that effect. So again, the nitrate treatment maintain redox potential the highest, above what otherwise will be in the native soil or in the iron oxide treated one. But we can see that the one that we're treating with sulfate actually deepen that redox potential significantly. And if we look at that specific date, we can see again those differences, and those differences are significantly different. And we can see that we can generate that redox ladder, and again that the sulfate treatment, meaning abandonment of sulfate, result in a much larger decrease of redox potential under same condition for that same soil. We have to tweak a little bit and add some substrate to that system in order to jumpstart some of the processes in the gray sandstone. And we see that after 7 days, again, we were able to generate that same thing with the nitrate maintaining a high redox potential above what the control soil will do, and that the sulfate addition actually result in a much lower redox potential, which again, the lower the redox, the more destabilizing those minerals that actually are stable in oxidizing conditions. We further looked, we kind of tweak a little bit the incubation system so that we can actually measure some of the nitrate. And we look here at two treatments. We look at incubation study where we added both nitrate and sulfate, or that we added nitrate only. And this is the brown sandstone. We do see the same differences in that redox impact, meaning the treatment that have that sulfate here in addition to the nitrate indeed maintain a much lower redox potential. But we also see that there is a difference as far as nitrate consumption over that incubation period by the two treatment, meaning that the treatment that actually have both sulfate and nitrate, we see that more nitrate is being consumed than the treatment that have nitrate only. And of course, the first thought was, well, maybe that system is just sulfate limited, just as a nutrient, and this will explain it. So we repeat that whole thing in an open jar system. So this is an aerobic system, unlike the anaerobic one that I just showed you. And we can see that in the aerobic system, we actually don't see any difference in nitrate consumption between the two treatments. So at least in the aerobic system, it suggests that sulfate does not increase nitrate consumption as it does in the anaerobic one. And again, we're still trying to figure out what exactly that will mean. But again, if we're just comparing this in a different way to just show those differences, we can see that, again, in an open system, we don't see any different in nitrate consumption, where in that closed system, the anaerobic incubation, we do see that nitrate only is actually not consumed as much as if we add sulfate to the system. So again, this suggests that sulfate stimulates some of that nitrate consumption, and of course have some impact in microbial activity and so on and so forth, that again kind of drive the chain of this redox processes and conditions. So again, to just summarize, we do see that redox conditions affect the total dissolved solid levels, as we saw in the field study, and those high TDS were associated with low redox or anoxic saturated, water saturated conditions. And we see that the abundance of terminal electron acceptor do affect and maintain a lower redox potential in the soils. And we do see higher decline in nitrate in the presence of sulfate under anaerobic conditions. And that all lead us at least to some kind of a working hypothesis that reductive dissolution do play a significant role in maintaining those high TDSs. And the greater impact is that, and again, we think that those soil that are made out of just a bunch of rocks and have very limited soil development stage might be missing some of those steps, and the fact that sulfate is ubiquitous to those mining activities, and we have elevated sulfate in those system and environment, that may, between those two, those driving factors, we may result in a lower redox potential that we will expect otherwise from just environmental or moisture condition to occur in a much more equilibrated, well-developed soil. I would like to, at this point, just to acknowledge people who were with us on the study, the funding agencies, and I will conclude at this point. Thank you.
So thank you, Amir, for a very interesting presentation.
Let me see how I give you control back here.
And now it's time for presentation. Now it's time for questions, sorry.
I have a question again.
Yeah.
Mikhail, you can ask.
Go ahead.
Okay, so that was really interesting. Thank you very much. So it seems that under anaerobic conditions, so there's a nitrogen sulfur co-limitation of these microbial processes. And now, to me, the question is, how long during the year, so do we have anaerobic processes in these soils, especially in the compacted soils, you know? There, when it's wet also, we will have lots of anaerobic microbial processes. And so to me, that's I think that's really key to understand these what you did here at a small scale.
Yes, great, Michael. And in fact, stepping into this whole study, the fact that we are talking about, you know, mostly the concern is that we actually do not have enough of a clay-sized material, and we have too much of a rubble gravel type material, and we do have, and I don't know if you paid attention to the first picture on this whole presentation. This was for the, if I can share real quick this, if you don't mind there. Let's see, okay. So we do see, do you guys see that?
Yeah, yeah.
Okay, so you can see that if anything, the least of concern should be with excess moisture and development of anoxic conditions. I mean, that's almost like ludicrous. We actually have too much of an excess infiltration there, and we, you know, the moisture is limiting, and to consider anaerobic condition development is almost preposterous to even think in such system that actually it's just a pile of rock for all intent and purpose. And let me stop sharing here. So you're absolutely right. So again, this is just compounding that intriguing issue, is that yes, we will expect those are the texture of the soils do not suggest that we will have any issue with infiltration, do not suggest that we will have any development of anoxic conditions. We don't have a lot of organic material to begin with to suggest that there is some explosion of activities. Yet, we see really low redox potential numbers that again kind of led us to think, hey, what is going on here? With this moisture, we will not expect such low levels in just regular soil, if you will. And it just might be that we have a lot of sulfate in those systems. And again, this is just a given, giving all the impurities of pyrite and isosulfur minerals that are in the system, that high levels of sulfate is a given in those systems. And it just might be that we're also missing some of those redox steps. So if we don't have a well-established, free iron or manganese oxide phases, then we're kind of have this broken. So if we're consuming the nitrate and the oxygen, you're kind of falling all the way down to the next best thing. And if sulfate is there to just save the day in a sense, then you'll end up. So again, it's mind-boggling also from just thermodynamic perspective, the fact that we can actually drive redox processes, meaning to have microbes somehow prevail with excess sulfate and use a much less efficient terminal electron acceptor compared to a control so the control should have provided that supporting level. But yeah, so again, it's to us, it's very interesting, and we don't have all the answers.
So I would have two similar questions. So basically you said you compare graded and ungraded or compacted or non-compacted surface. So do you see, maybe you mentioned it, I miss it. Do you see any difference between this compacted and uncompacted treatments in terms of this behavior, higher solvents in a more redox environment?
Absolutely. So, again, with this whole conceptual framework, we were expecting that the compacted plots will have higher moisture content, and in fact, we do see some inundation of that system. So we have high level of moisture there, but in fact, the highest level of iron and manganese, soluble manganese and iron. And again, we're looking at soluble, meaning we're not even looking at total. So we're looking at that filtered sample under the 0.45 microns. So I have to admit that our highest levels actually occur in the non-compacted as opposed to the compacted. Now, in the compacted, we expected much higher levels of moisture and those to be maintained at much lower length, but we don't see that system seem to be in equilibrium as far as are concerned with total dissolved solids. It reached some kind of an equilibrium that does that constant state of high moisture do not result in high levels of TDS. And in fact, those fluctuation in the drained, in the non-compacted, which is more drained, the drainage is much better, we do see those fluctuation, and that's mostly where we reach those high levels. And I don't know if you remember that those graph with the data points, but it's the brown sides that actually reach those highest, but you're absolutely right in the sense that I didn't differentiate between the compacted and non-compacted, so you couldn't have seen that there. But it is the non-compacted that actually reach those higher values, and not the more expected redox impacted compacted one. And we believe that it's just some level of equilibrium, if you will, that maintain those lower level of total dissolved solid. And again, it's the total dissolved solid that kind of keep everyone's awake at night as far as those operations and for them meeting those obligations. But we didn't, everything is opposite to what the expectations will be. So yes, you will expect the compacted to have a higher level. They do maintain high threshold level, but those peaks occurring actually in that non-compacted.
Yeah, and this, I just want to clarify. So you just dump it, you compact it or not, and then you start your measurement. So this is very initial, or this is...
No. So in 2005, the site was established, and it was established in a way to compare two things: the different substrate, the gray versus the brown, and within each one of them, compacted versus non-compacted. Well, the compacted is a number of passes that they run with the dosers as they put that material and level it off. You know, they're mostly engineers, so they just look to laser and to put it straight and to compact everything so nothing is moving. And over the years, you know, they figure out that maybe when we try to actually develop soil, that's not the great practice. So the non-compacted was actually with the minimal. So I think the difference was between 2 and 6 or 8 passes of that dosers. So the resulting landscape is that within that non-compacted, you have a much more variable micro landscape environment. But again, this was established in 2005. We came in 2017, so 12 years after. Okay.
Yeah. So can it be so that in compacted sites which have higher moisture, there was already a larger leaching of something, and that's why it shifted?
It might be that you're absolutely right. And let's put it this way. I think that if we were to do it, you know, instead of the 3 years from 12 to 15, if we were done it from 1 to 3, I think that we would have fallen to the same conceptual framework, that it is the acid promote dissolution that govern that system. And there, of course, will be a larger leachate from both systems, but it certainly might be that, you know, we came late to the party in a sense on the compacted one, where a lot of it has already been leached. Yeah, it's certainly a possibility.
Okay, so thank you all speakers for their presentations, and now it's time for Jan.
Okay, so
final.
I would like, there is still breakout room after this session. However, I would like to thank you all for, you know, coming. It was really amazing to see people all around the world, basically, you know, meeting here and across time zones. And I was really, I think, more diverse than an ordinary conference, I would say. And so I was really happy we meet here. And I was really enjoying this exchange of information. I think if you don't receive the abstract book, you should receive it early next week. And there is a YouTube link, and I guess this can be accessible even after the conference is closed. So if some of your students or your colleagues might be interested to see some of the presentation and get back to that, I think they still can be seen on YouTube. And I would like to thank all the people who were coming. And actually, if I might ask you, if you can turn on your camera as much as possible, who are here. So I would actually like to make a thing which is quite common on international conferences, and we forgot to do it yesterday, and that will be a group picture. So if you can turn on your camera as much as possible, and I will make a print screen of our faces, and I will distribute it to you as a group picture from the conference. Yeah. So there's someone else who is able to turn camera on. Okay, so that seems that's it. So I'm just making a print screen, and I hope that will work. I just save it to, yeah, went very nice. So we will send you the group photo for the conference, so at least we keep up one tradition which is common in international conferences. So once again, thank you for coming. There is a breakout rooms after this session. I will not keep any closer speech after that, but enjoy the breakout rooms. I really enjoy the meeting, and I close the conference by this one. So, but still the breakout rooms are open, so we can enjoy that and discuss with speaker of previous session if you have any further question.
Thank you, Jan. I will open the breakout rooms for 15 minutes. So,
and Jan, here is another question about summer school. So how it will be organized and so on.
Sorry. That's important point. So basically, I also would like to invite everyone who was signed for summer school. If you get interested during the conference and you see some of your students might be interested in summer school, there's still possibility to sign, write Marketa, write Adela. I will already send the email about that, which in the program. So write Adela Kotabova, and she will still include the people to the mailing list for the Zoom for the summer school. We will distribute the link. So probably early morning for the people. So, and we are going to organize a summer school. We already distribute the information about the timing to the people who signed for summer school. And the summer school will be organized from Monday to Thursday in 2 teaching blocks. Teaching blocks will be from 11 to 12:30 and from 2 p.m. to from 12:30 to 2 p.m. Yeah. No. From 11 to 12:30 and from 1 p.m. to 2:30 p.m. So this actually arrangement, we look on the time zone of people who